My request to GC Nanda sir also please to come on the stage.
दधातु दयोः शान्तिः अन्तरिक्षं शान्तिः पृथ्वी शान्तिरापः शान्तिरोषधयः शान्तिः वनस्पतयः शान्तिर्विश्वे देवाः शान्तिर्ब्रह्म शान्तिः सर्वं शान्तिः शान्तिरेव शान्तिः सा मा शान्तिरेधि यतो यतः समीहसे ततो नो अभ्यं कुरु शन्नः कुरु प्रजाभ्यो अभ्यं न पशुभ्यः ॐ शान्ति शान्ति शान्तिः.
Now my request to our dignitaries to please take their respective seats. It's an immense honor for us to have such esteemed personalities today with us. Now, my request to Honorable Chandra Mohan sir and his team members to mesmerize the surroundings with the beautiful bhajan sounds as Prabhu tum anu se bhi sukshma ho aur Prabhu tum gagan se bhi vishal ho. Over to Chandra Mohan sir. Please welcome him with a huge round of applause.
ओम. यहाँ पर उपस्थित आप सभी प्रबुद्धजन, सभी अतिथिगण, सभागार में उपस्थित आप सभी साइंटिस्ट वृंद, विद्यार्थियों को मैं चंद्रमोहन मिश्र प्रणाम करता हूँ. एक बहुत सुंदर से भजन जिसके बोल हैं प्रभु तुम अणु से भी सूक्ष्म हो, प्रभु तुम गगन से विशाल हो, मैं मिसाल दूं किसी और की, दुनिया में तुम बेमिसाल हो. प्रभु की महिमा हो तो, प्रभु की महिमा हो तो पत्थर पानी पर तिर जाता है. एक हवा का झोंका भी भीषण तूफान उठाता है. उनकी मर्जी अगर ना हो तो कश्ती साहिल पर डूबे, उनकी कृपा हुई तो तिनका नदिया पार कराता है. प्रभु तुम अणु से भी सूक्ष्म हो, प्रभु तुम गगन से विशाल हो, प्रभु तुम अणु से भी सूक्ष्म हो, प्रभु तुम गगन से विशाल हो, प्रभु तुम अणु से भी सूक्ष्म हो, प्रभु तुम गगन से विशाल हो. मैं मिसाल दूं तुम्हें कौन सी, मैं मिसाल दूं तुम्हें कौन सी दुनिया में तुम बेमिसाल हो प्रभु तुम मणु से भी सूक्ष्म हो प्रभु तुम गगन से विशाल हो। हर दिल में तेरा धाम है और न्याय ही तेरा काम है।
हर दिल में तेरा धाम है और न्याय ही तेरा काम है।
हर दिल में तेरा धाम है और न्याय ही तेरा काम है और न्याय ही तेरा काम है। सबसे बड़ा तेरा नाम है सबसे बड़ा तेरा नाम है। जगन्नाथ हो जगपाल हो मैं मिसाल दूं तुम्हें कौन सी मैं मिसाल दूं तुम्हें कौन सी दुनिया में तुम बेमिसाल हो प्रभु तुम गगन से विशाल हो प्रभु तुम गगन से विशाल हो। और हम सभी अपने जीवन में साधना को प्राप्त होते हैं, साधक हैं अपने अपने क्षेत्र के। तुम साधकों की हो साधना या उपासकों की उपासना।
तुम साधकों की हो साधना या उपासकों की उपासना।
तुम साधकों की हो साधना या उपासकों की उपासना या उपासकों की उपासना। किसी भक्त की मृदु भावना किसी भक्त की मृदु भावना या किसी कवि का ख्याल हो या किसी कवि का ख्याल हो। मैं मिसाल दूं तुम्हें कौन सी मैं मिसाल दूं तुम्हें कौन सी दुनिया में तुम बेमिसाल हो प्रभु तुम मणु से भी सूक्ष्म हो प्रभु तुम गगन से विशाल हो प्रभु तुम गगन से विशाल हो। मिले सूर्य को तेरी रोशनी को तेरी रोशनी, खिले चांद में तेरी चांदनी, सब पर दया तेरी पावनी। प्रभु तुम तो दीन दयाल हो, प्रभु तुम तो दीन दयाल हो। मिले सूर्य को तेरी रोशनी, तेरी रोशनी, तेरी रोशनी, तेरी रोशनी। मिले सूर्य को तेरी रोशनी, खिले चांद में तेरी चांदनी, खिले चांद में तेरी चांदनी, सब पर दया तेरी पावनी, सब पर दया तेरी पावनी। प्रभु तुम तो दीन दयाल हो, मैं मिसाल दूं तुम्हें कौन सी, मैं मिसाल दूं तुम्हें कौन सी, दुनिया में तुम बेमिसाल हो। प्रभु तुम अणु से भी सूक्ष्म हो, प्रभु तुम गगन से विशाल हो, प्रभु तुम गगन से विशाल हो, प्रभु तुम गगन से विशाल हो।
Thank you, sir, for such a phenomenal performance. It is always a blessing for us to hear. Thank you once again. Please give him a huge round of applause to him and his entire team. Thank you once again, sir. Now, we'll be heading towards our first technical session of the day 2, themed as Unveiling the Ayurgenomic Approaches for Attaining Holistic Health. In this session, we will explore the fascinating intersection of Ayurveda and genomics and will shed light on the potential of integrating these disciplines to unlock new frontiers in holistic healthcare. The theme of this session emphasizes the integration of Ayurveda, a centuries-old holistic healing system, with the groundbreaking field of genomics. Ayurveda, with its profound knowledge of individual constitution, balance, and preventive care, provides a holistic approach to well-being. Genomics, on the other hand, offers insight into our genetic makeup and its impact on our health. By combining the ancient wisdom of Ayurveda with the cutting-edge science of genomics, it will open the door to personalized, precise, and holistic approaches for complete health and well-being. Today, renowned experts will share their research findings, insights, and practical applications, highlighting the immense potential for this integration in achieving holistic health for individuals and communities. In this session, we have 4 renowned speakers from different parts of the world, and to chair the session, we are delighted to have Dr. Narendra Bhatt sir and Professor Gopal C. Nanda sir. Regarding our virtual session chairs, Dr. Narendra Bhatt sir has presented Ayurveda on several international platforms on behalf of the Union government. He remained to be a member of project on indigenous knowledge for health under Global Research Alliance funded by the World Bank. Presently, he is serving as an Ayurveda consultant in NAM University. He is closely associated with Indian Association for the Study of Traditional Asian Medicines. Sir has already been honored with several international and national awards. Please welcome such an eminent personality with a huge round of applause. My request to sir to please join us on the stage as session chair. Please welcome him with a huge round of applause. Our second session chair is Professor Gopal C. Nanda sir. Sir is a renowned Ayurveda Acharya and has been awarded with Rajvedya Award from Governor of Assam in 2013. He is presently serving as the Chairman of Empowered Committee in Ministry of Ayush. He is also the Director and HOD of Kaya Chikitsa in Prabodh Ayurved Medical College and Research Center, Lucknow. It is an honor for us to have such a resplendent personality today with us. Please welcome him with a huge round of applause. My request to GC Nanda sir to join us on the stage as session chair. Please keep applauding for him. Once again, we welcome the conference chair of the technical session. Please give a huge round of applause to both of our session chairs. Now, I request our session chairs to please introduce the first speaker of the event, Dr. Kalpana Joshi ji.
Hello. A very good morning to all of you. The subject is of very interest and I am very happy that our first speaker, initially I know her for many years, when she began her activities in the field of Ayurveda and genetics long back at the Interdisciplinary School of Research in University of Pune. Now it is Savitribai Phule University, right? So she is a very, working as the professor and the head of the biotechnology department at Sinhagad College of Engineering, which is affiliated to the SP University. She is recipient of the M.M. Sharma Award for Research in Science and Technology. More than reading her things, what I would like to suggest is that she's a very dedicated researcher as I know her. And some of the very senior persons with whom she has worked, I firmly believe that without Kalpana's contributions, those work would not have been completed and fulfilled. And that is very significant of you, Kalpana. And she has been chairman of the Board of Studies. She has been holding several positions in the university. And with these words, I ask Kalpana to come and share her experience and views on this important subject. Kalpana.
Good morning. At an outset, I would like to thank Dr. Bhatt sir, who really has given very warm introduction and reminded me of those days when I was just very active and young and just began with my Ayurveda research. Thank you, Professor Nanda, for chairing our session. I thank organizers and give my pranam to Swami ji as well as Acharya ji for inviting me here for this session. So I would like to begin with some introduction to Ayurveda biology. So students of Ayurveda and many other disciplines are here. So I would like to inform them that I began my research in Ayurveda the way you are doing or starting your career. So with some stint of pharmacogenomics abroad and my PhD degree in molecular biology, I joined School of Health Sciences, and then I was introduced to Ayurveda. Till then, Ayurveda, I knew only about taking ginger for some cough and this thing. So I had no idea about Ayurveda. But that was the time where I was introduced to many Ayurveda experts. I would like to name Dr. Bhatt was there, Dr. Vaidya Nanal, and many more colleagues, Dr. Nesri, Dr. Tanuja, and many others were there who really contributed some knowledge about Ayurveda. And then I got to read some of the basic books on Ayurveda, and that is where I got introduced to basic principles of Ayurveda like Prakriti and many more. While I was discussing with my Ayurveda experts about Prakriti, and as you know, I have some basic background of pharmacogenomics work, it really emerged in our mind that Prakriti is so special to every human being. It is very, very specific to humans. And then Ayurveda physicians, they prescribe their drugs, diets, and many things based on the Prakriti of a person, which doesn't change after you are born. So there could be some vitiations of Prakriti which could be taken care of by giving some drugs or methods like Panchakarma. So that is where we thought that Prakriti could have some genetic connotation, and that's how we began with our discussions. And then some of the work. May I have the next one? Next slide is not moving. Okay. This was my first impression about Ayurveda, where we see that there are 3 data sets about body, where you have Prakriti and other things, then diseases or various symptoms are mentioned, and then there are drugs or say therapies for combating the diseases. So there are so many basic sutras, and there are 3 books, Charak Samhita, Sushruta Samhita, and Vagbhat classic commentaries there, and I was able to get some knowledge because there were no English or Marathi books on these, but then some of the contributions by Professor Valyatan gave us enlightenment of these 3 books, Brihatrayi. So I am being molecular biologist, I am totally reductionist person, and Ayurveda being holistic science, I tried to compare Ayurveda science with reductionist molecular biology, and you see that the molecular biologists, they go from organs to tissues to cells to molecules like DNA and RNA and proteins, and here is the Ayurveda which talks about holistic, which is holistic science of life, and it talks about Pancha Mahabhuta and Pancha Mahabhuta being in you, or say microcosm to macrocosm. So this is where we began to integrate Ayurveda with molecular biology. And then, I'll just skip this slide. And then later on, when the concept of applying genomics or studying genomics of Prakriti emerged in our mind, we started discussing it. And then we thought that why not do some small study where we could find what is the genetic basis of Prakriti. Because if you look at this picture, Prakriti is predictive, it is like you are able to prevent some of the diseases if you follow the Prakriti rules, and then the medicines could be personalized based on the Prakriti. So Prakriti is almost like pharmacogenomics or personalized medicine. So we thought of correlating the two and began with a new word called as Ayurgenomics, where we tried to integrate Ayurveda concepts with the molecular concepts, or say molecular biology. And with this integration, we were able to show that Prakriti has genetic connotation. So our earlier work on HLA genes, it told us that HLA genes, they are correlated with some of the Prakriti. So some HLAs are correlated with Kapha Prakriti, Vata Prakriti and Pitta Prakriti, and then those HLAs were giving us proneness to some of the diseases like arthritis. So this was the first paper showing that Prakriti has genetic connotation. And then we explored in many ways, like Prakriti, Kapha Prakriti has slow metabolism, Pitta Prakriti has fast metabolism. So we thought, why not use them on drug metabolizing enzyme gene polymorphism. Then we looked at those gene polymorphisms and then found, this time we were not surprised, because we found that drug metabolizing enzymes, which are fast metabolizers, most of those polymorphisms were shown in Pitta Prakriti. The slow metabolizer polymorphism, they were shown in Kapha Prakriti. So once again we proved that Ayurveda Prakriti has some genetic connotation. And then came a nationwide interest in Ayurveda Ayurgenomics, or say Prakriti genetics, and that is where Ayurveda biology emerged. Professor Valiathan, in his mighty paper, which was written at National Science, and then this paper, then he invited us to contribute various projects and various new ideas to Ayurveda biology research. And we were the ones going and presenting our work on genome variations in different Prakritis. So that is where it was picked up by scientific Prime Minister's scientific advisors, and we were given big funding to carry out this research at the nation across. And we had collaborators from right from down Bangalore to Delhi, and CCMB and Indian Institute of Science and many more institutes were involved in this project. So there were several projects which were conceived to check the basic principles of Ayurveda. We all know that there is lot of investigations, or there is lot of research in pharmacology, Dravya Guna Vignan, and finding out new drugs from Ayurveda. But there was hardly any research on basic fundamental principles of Ayurveda, like whether Prakriti has genetic connotation, whether Panchakarma, they give any immunomodulation, what are bhasmas, whether there is any nanoscience involved in bhasmas. So such fundamental principles were explored under the program of Science Initiative in Ayurveda, under Ayurveda Biology, which was mentored by Professor Valiathan. I was the principal investigator in genome variation analysis and expression profiling of dosha Prakriti. So this project was, we recruited subjects with different Prakritis. So as you all know that we have to go on finding out the Prakriti, and as we know that the two Vaidyas may not match their outcome. So we developed a software called IUsoft with the help of CDAC in Pune, and then this software was used for the detection of Prakriti, as well as eminent Vaidyas, they carried out Prakriti. And when two of the Prakritis matched, like major of Kapha and minor of Pitta and Vata, so such when scores were developed, and then we isolated DNA and then carried out whole genome SNP as well as gene expression analysis of these people. So we had more than, it was like 1000 genome, because we had more than 1000 people with 3 different major Prakritis, and we analyzed them for whole genome SNP analysis as well as whole genome expression analysis as well as epigenetic analysis. These are all 3 hallmarks of genetics. And then we found that Prakriti, or say here, like we found that Pitta is an expression of Agni, and the gene like PGM1, which is very important in energy production, was found to be associated with Pitta Prakriti. So like this, there are many more results, and several genes we found which are correlated with particular Prakritis, giving them a particular qualities. So this paper was published in Scientific Reports, and it has got good citations. Then another work is, we all know that our genetics doesn't change. Our Prakriti is decided at and it never changes after we are born. But there are vitiations of Prakriti. So what could happen if there is some disease precipitated because of the vitiation in particular Kapha, Pitta and Vata? So we thought that these changes cannot occur in your sequence, or say DNA sequence, but these could happen in your expressions. So the change in expressions, they could be because of the methylations or epigenetic changes. So the challenge was to find out what are epigenetic changes or methylations within the Prakritis. So we assessed 3 Prakritis, Kapha, Pitta, Vata, and we studied their global methylations in these DNAs. And then we found that there are certain methylations which were common in Kapha Prakriti, some of them were common in Pitta and say Vata Prakriti. And then we also hypothesized that when there is some particular vitiation or dosha, then those methylations may be increasing or decreasing. So we had a paper on DNA methylations in phenotype specific or say different Prakritis. Recently, with our team in health sciences, we tried to find out a solution for COVID, because in Corona that we all know that who will suffer with cytokine storm and who will succumb or who will recover and get back. So that was very difficult to find out what is the prognosis of Corona in given patient. So we thought that Ayurveda can play a role in finding out this, and then we tried to find out whether Prakriti has any role in giving prognosis of Corona or consequences of Corona, and this project is ongoing. So while we were studying genomic approaches in Prakriti, and we all now have almost know that there is some connotation genes involved in Prakriti. So how does disease affect us, or how does any Ayurveda treatment it affects us, or it cannot make any changes in our DNA sequence? So how does it make the change in our system? So that is where we thought of doing a whole system approach, and the disease chosen was asthma, because asthma has lot of gene environment interaction. So you may have certain genes because of which you may have certain proneness like obesity, diabetes and so on, but 80% of the environment contributes to developing those genes into disease genes. So in this project we thought of looking at epigenetics of asthma when it is treated with various Ayurveda therapeutics. So we took into consideration asthma patients, various treatments planned for them with according to their Prakriti, or there are 2 types of asthmas, Kaphaja asthma and Vataja asthma. So accordingly the treatment was planned. The whole project was initiated because I came across this paper which said a patient with asthma seeks medical advice in 1928, after 100 years, and then 2012. Okay, so this was published in NEJM, or very prestigious journal in medical science. And then they said that there is lot of going on in developing the pathogenesis or finding out molecular mechanisms under pathogenesis, various cytokines and IgE and whatnot. But there is hardly any change in treatment of asthma. So most of the treatments given for asthma, they are bronchodilators. So they are symptomatic treatments, like they dilate your bronchi and you are able to breathe. There are no treatments which removes the root cause of the disease. So we thought, why not look at Ayurveda, whether it really removes root cause of the disease. And you will be surprised to see our result. Like yesterday, so Amiji also mentioned that Ayurveda removes root cause of the disease. So we'll come to that now. So the whole treatment given to the patient, this was again funded by Office of PSA. It was with Tiyak Ayurveda Mahavidyalaya in Pune with Dr. Tanuja Nesri as investigator in this. So we decided Ayurveda treatment regimen for the patients. Now patients could have Vataja asthma or Kaphaja asthma. We had lot of brainstormings with various eminent Ayurveda acharyas in Pune. And I will tell you that I am modern medicine or say molecular biology person, and when they said that asthma is disease of stomach, you know, I laughed and laughed and laughed, because we all know that asthma is disease of lungs and bronchi, and they said asthma is disease of stomach. So I just couldn't control myself, and how it is going to be asthma disease of stomach, I had no idea. But then we had lot of discussions, and they planned treatments for Kaphaja and Vataja asthma. And then I tell you all, like after you will see the results, the way it is modeled through molecular biology, and you will see that it is, I started believing that yes, it is disease of stomach, you know, not just disease of lungs. So for Ayurvedic discipline, this might be of interest to you. That's why I have kept this slide here. So patients were diagnosed whether they have with modern medicine bronchial asthma. Then they were classified as Kaphaja asthma or Vataja asthma. And the treatment regimen differed for Kaphaja asthma as well as Vataja asthma. So it passed through the steps like Pachan or say improving Pachan. Then Snehan was there, Swedan or steam was there, then Sneha Pan was there. Then for the patients who had good Bala, they were given emesis or Vaman, and then there were Shaman drugs and diet, and then after Shaman drugs they were given Rasayana or immunomodulatory. So this was the regimen for every patient according to his Prakruti and the type of disease. After, during this treatment, like we tried, we analyzed cytokine profiles as well as DNA for methylation before the treatment and after the treatment, and post treatment 6 months. So the treatment followed for 6 months till Rasayana, and after Rasayana we just interacted with patient to know their status for remaining 6 months. And we carried out cytokine analysis, multiplex cytokine analysis, and at a given time 21 to 48 cytokines were analyzed, and you will see that this was very surprising that various cytokines which are inflammatory cytokines, Th1, Th2 cytokines, those cytokines were brought almost to the, if you see, level of the healthy control people. Like you can see the reduction in IL-4. So similarly, various TNF alpha, you can see it is reduced a lot. So most of these inflammatory cytokines, they were reduced to the healthy control subjects, or they were highly significantly reduced because of the Ayurveda treatment. Pro-inflammatory cytokines, IL-1, beta, 6, 8, they were also reduced very significantly to the healthy level. And then immunosuppressive cytokine like IL-10, you will see that this was the healthy control, this was before treatment, and then it was increased after the treatment. So immunosuppressive cytokine was more, which suppressed the inflammation. So many of the cytokines, they were modulated because of this whole system treatment by Ayurveda. Now if you look at this graph, like you will see that eosinophil count was significantly reduced, and IgE, which is hallmark of allergic asthma, so IgE is very significantly reduced. Up till now, there is no treatment except steroids which removes or which reduces IgE or allergic antibody. So all other treatments, they do not at all have any change in IgE or allergic antibody. So here, like we saw after the treatment, the root cause, which is allergic antibody, is removed from the body. And there is one huge mechanism because of which this IgE is removed. Now to know this mechanism, we went through methylation studies where we carried out epigenetic analysis or DNA methylations before the treatment and after the treatment, and then you will see this was just recently published. So then you will see that there is differential methylation in various transcription factors which are involved in immune processes. So it does tell us that Ayurvedic treatment has removed the root cause. The IgE is reduced down, which is responsible for allergy. So IgE is reduced down because of certain changes in cytokines like cytokine 4 and all. And those who know immunology, they know the B cells, they are responsible for producing IgE. So B cells, they produce IgM and then they switch to IgE in allergic condition. But because of the Ayurvedic treatment, the B cells, instead of producing IgE, they have switched to producing IgG. So this is called as class switch. So this class switch has happened because of the methylations of various regulatory transcription factors, because of which there is total shift in cytokine productions as well as allergic antibody production. So this is where I think this is the first time we have shown that Ayurveda removes root cause of the disease, and it has really reduced down IgE antibodies in diseased persons because of the whole system treatment. Coming to this, like we have been exploring various approaches. We tried using molecular biology, genomics, gene expression analysis for studying effect of various drugs, like how Arjuna works in heart disease, how many more drugs they work in various diseases through gene expression analysis. If you just scroll through Google, you will find many publications from our group on gene expression studies. In similar line, we developed an in silico approach called as network pharmacology or chemogenomics. So before you actually begin with any molecular biology, you could check it at in silico level by having various networks. So this was the one which was from Dr. Patwardhan's group on Triphala. Like we know that it's a great combination of 3 fruits and then it works at various targets. But then what happens, or how do you show that it works at various targets? So the network analysis was carried out, and Triphala was also shown to be involved in cancer activity. In similar line, we carried out network analysis of Tinospora and gingiber or ginger, and then found that it has got lot of targets in human breast cancer, and it was again later found out to be effective in triple negative cancers, or it was validated through real experiments. So such in silico approaches are useful in finding out mechanism of action of Ayurvedic drug. So with our Harvard collaborators, we have had good work on comparing Ayurvedic medicines for their mode of action in silico approaches. Then there is one huge contribution of Ayurveda, Rasayana Chikitsa, or it is also called as rejuvenation. My colleague Asmita will be talking about Rasayanas. But then as a cell biologist, molecular biologist, we all now know what are the functions of Rasayanas and how they are used by Ayurveda. So it is cleansing srotas and dhatu nourishment and many more. I need not explain all these things. So prevention of diseases, and they also contribute to longevity. So such activities of Rasayanas, it came to our mind whether they act on our stem cells and strengthen the stem cells. When it says it strengthens the dhatus or say tissues, and then we put forth our hypothesis that Rasayanas may act on stem cells and may able to strengthen stem cells, proliferate them, and also help them differentiate. So we chose the Rasayanas, which are, we all know that Medhya Rasayanas are there, Hrudya Rasayanas are there, general Rasayanas, like we had yesterday's story of Amla, So we chose these rasayanas and then applied them to the mesenchymal stem cells, and then to our surprise, like we found that these stem cells, they had elongated life. Like otherwise, if you could grow stem cell till say 5th passage, 6th passage, in presence of rasayanas, we were able to grow them till say 14th passage. It means they helped in maintaining the stem cells or also proliferating the stem cells. So with various rasayanas, we could see that the stem cells are able to better proliferate when you treat them with rasayanas. So rasayanas do have some rejuvenating activity because they are able to strengthen our stem cells or proliferate our stem cells. So we showed that herbal or say rasayana preconditioning, it delays senescence in mesenchymal stem cells and allow them to better proliferate in presence of rasayana. Then we studied if they are able to do any action in differentiation. Now stem cells have ability, they are pluripotent, multipotent, totipotent and many more. So they are able to differentiate into say bone tissue or say neural tissue or any other tissue, so muscles and so on. So stem cells are able to differentiate into adipose cell and many more. So we tried to look at various differentiation potential of these rasayanas, and we found that many of these rasayanas, as the name suggests, Medhya rasayana or say Rudya rasayana, they are able to differentiate into those kind of differentiated cells. So this is one of the examples I have given where rasayana, when they were treated, we found that there is lot of calcium which is taken up and then there is osteogenic differentiation. So Piper longum was one of the best rasayanas which could take up calcium a lot and also differentiate cells into osteogenesis. In similar line, now various rasayanas are being tested for neurogenesis or say cardiomyocyte development and so on in our lab. Recently, Ayush Ministry had invited papers for their 100th Man Ki Baat issue, and then I was invited to contribute to this issue of a journal published by Ayush, and I'm really happy that with my colleagues and with lot of discussions, like our group keeps moving forward. You know, we worked in genomics, then we went to network pharmacology, then to stem cells, and now we are into using artificial intelligence as a tool to improve and modernize Ayurveda treatment. So there are a lot of smaller elements of the three data sets in Ayurveda. So we could use those data sets and then bring a particular kind of structure where we could say increase or reduce particular dosha through the diet and other regimens, and then we are able to bring back the homeostasis. So we are going to explore such approach, artificial intelligence approach, using various Ayurvedic treatment data and then use it to show or see if there is homeostasis or the balance obtained through this. So we keep developing various concepts, and even the students, they carry out small projects. Like one of the projects was like transcutaneous absorption. We always do massage to babies, so whether it really works or no. So we actually carried out this work with pediatrics department, and we showed that this is really, massage is helpful to the low birth weight babies and it helps them, preterm babies. In similar line, we analyzed ghee, and we showed that the ghee which is prepared with Ayurvedic method, like we culture it, make it dahi, churn it, and then bring up butter and then boil that butter to convert it to ghee. So if we use this process, which is Ayurvedic process of making ghee, then there is lot of docosahexaenoic acid, which is called as omega-3 DHA fatty acid, polyunsaturated fatty acid. And in vegetarian diet, there is hardly any DHA, because what we have is ALA. So it is rich in fish and eggs, but we don't eat that. So where is DHA in our diet? So DHA is in ghee. So if the ghee is prepared in Ayurvedic way, there is a good 30% rise in DHA which is available to us. That's why like all our traditions like giving ghee or eating more ghee during pregnancy, delivery, so they are good because DHA is, you are able to get DHA only through the ghee, and then DHA is good for the brain development. So there are so many concepts in Ayurveda, like fundamental concepts like rasa, virya, vipaka, dhatu. So all these need to be explored, and we need to carry out more research in exploring these fundamentals. I can give you one small example. There you might have learned some biochemistry. So rasa, virya, vipaka are major three things to assess the drug in Ayurveda. So for example, sugar has madhur rasa. So what could be the rasa for lemon or lime? So Ayurveda says the lime, even if it is sour, the rasa and vipaka is sweet, madhur. So if you see, if you apply your knowledge of biochemistry to it, the citrate which is there in limes, it never undergoes direct metabolism to carbon dioxide. Citrate is turned back to glucose through gluconeogenesis, and then it undergoes glycolysis. So like citrate is converted to sugar, that means the vipaka is sweet, isn't it? So such kind of concepts need to be explored and find out what Ayurveda, always believe what Ayurveda says and try and apply. Otherwise any biochemist would say ki lemon is sour, so how Ayurveda said, Ayurveda has done mistake in saying it is madhur vipaka. But if you relate it, citrate is always converted to glucose in our body. So the vipaka is definitely madhur. So you are able to apply so scientists may not have answers to many questions, but if we have to apply logic and explore science behind every concept which is mentioned in Brihatrayi or say Ayurveda, and then Ayurveda doesn't need anything to prove, but we scientists need some kind of logic or explanation. So our research can give us such kind of logic. Currently, I am a guest editor for this JAIM special issue on Rasayana, immunomodulation and regeneration. So if you have any contribution, please do contribute and submit your papers in area of Rasayana for JAIM. So thank you. I think this was our journey. So these are my mentors, Professor Valiathan, Dr. Bhushan Patwardhan, with whom I worked for 7 to 10 years, Professor Dr. Ashok Bhai, who always guided me in everything, and Dr. various colleagues, Dr. Bonde, Dr. Girish Tillu, Tanuja, Satyamurthy, Kondaya, Thangaraj, who all contributed to our research. Darshan Shankarji is also there. He always encouraged me in doing my stem cell research, and he had immense faith that stem cells will definitely respond to Rasayana, and that is how I started my stem cell work. And finally the Acharya ji and Swami ji, to whom I pray for giving us strength to carry out this research. Thank you.
थैंक यू मैडम, आपने जो पेपर आज पढ़ा इसीलिए मैं आपको स्टार्ट से कर रहा हूँ, क्योंकि आज आयुर्वेद के आंखों को अगर किसी ने खोला है तो मैडम ने खोला है। इसके बारे में मैं कुछ नोटिंग्स में किया है और हो सकता है इसको 2 मिनट आप सुनिए। मैडम, पहले आपने जो आयुर्वेद बायोलॉजी के बारे में कहा, एक ग्रेट माइलस्टोन फॉर द नेक्स्ट फ्यूचर जनरेशंस ऑफ आवर आयुर्वेदिक स्टूडेंट्स, क्योंकि ये बायोलॉजी जो कि कल भी हमारी इस डिस्कशन में आई थी, तो इसके बारे में मैं भी कहना चाहूँगा कि जो पर्सनलाइज्ड मेडिसिन आपने जिक्र किया है, आजकल मॉडर्न में वही चल रहा है, बट स्टैटिस्टिक्स के जरिए सबको कॉमन मेडिसिन दी जाती है। बट यू सी इन आयुर्वेदा न ही सर्वाणी शरीराणि व्याधि क्षमत्वे समर्थानि भवन्ति। सो ऑल द बॉडीज आर नॉट इक्वल टू एक्सेप्ट एनी काइंड ऑफ मेडिसिन्स। दैट्स वाई पर्सनल बात पित्त कफ का जो हम लोग आकलन करना शुरू करते हैं। दूसरी बात है कि जो आपने स्वस्थ लक्षण भी कहा, इज नॉट जस्ट ए सिम्टम, ये टोटल होल डिजीज को जो है डायग्नोसिस करने का। कोविड चल रहा है, कोविड का भी छद्म रूप अभी चल रहा है, थोड़ा मोड़ा कोई टेस्ट तो नहीं करते हैं, लेकिन थोड़ा छींक आती है, बुखार हो जाती है, तो इमीडिएटली वी आर टेकिंग सर्टन मेडिसिन आर सिम्टोमेटिक शुड बी स्टॉप्ड। ओके, दूसरी बात है आयुर्वेदिक जेनोमिक्स। ये सबसे बड़ा इंपॉर्टेंट टॉपिक आपने कहा, जिसके ऊपर भी कार्य हो रहा है, और ये नेक्स्ट जनरेशन में आयुर्वेद छात्राओं को, शोधकर्ताओं को भी नमन करता हूं कि आप लोग इसमें जुड़ जाइए, क्योंकि आयुर्वेद का शुक्र शोणित संयोगे जो भवेद दुषोत्कटः प्रकृति जायते स्तेन तस्यामे लक्षणम श्रुतुम। ये तो लिखा हुआ है 1000 साल पहले कि जब शुक्र शोणित स्पर्म एंड ओवम आर कंबाइंड टुगेदर दिस स्टार्ट्स योर दैट इज आयुर जेनोमिक्स। तो डीएनए वहीं से ही ट्रांसफर होता है। कैसे? जत जीवे जीव भागे बीज बीज भागे उत तप्तो भवति प्रकृति जायते ताना तेना शु लक्षणम शुणु। मीन्स व्हेन इट्स सेड क्रोमोसोमल डीएनए का भी जो डिविएशन होता है दैट डिपेंड्स अपॉन द स्पर्म एंड ओवम आल्सो। सो ये बहुत इंपॉर्टेंट टॉपिक आपने आज कहा है, बहुत ही इसलिए सराहनीय है। और जो मेध्य रसायन आपने कहा, ये मेध्य रसायन आप देखिए, मेध्य अकॉर्डिंग टू आयुर्वेदा 40 एज के बाद धीरे धीरे सबका जो है मेमोरी में डिस्टर्बेंस आती है, क्योंकि उससे पहले नौकरी हो सकती है, किसकी शादी हो जाती है, बच्चे पैदा हो जाते हैं, तो प्रॉब्लम आ जाता है। तो जब प्रॉब्लम आता है, इसलिए आयुर्वेद ने कहा कि आपको 40 साल पहले से ही शुरू कर देना चाहिए। पूर्वे वयसी मध्येवा युक्तो रसायनो विधि, ये आयुर्वेद का चरक का शौक है। अर्थात हम अगर रसायन थेरेपी करना चाहेंगे, नॉट इन ओल्ड एज, अगर हम अभी रसायन करना स्टार्ट करेंगे, ये मेरे को सूट नहीं करेगा, क्योंकि हमको पूर्व वयस में, यानी अगले 50 साल के लिए अगर हम इसको लें, एक प्रोजेक्ट लें, तो हो सकता है स्वस्थ भारत बन सकता है। पूर्वे वयसी मध्यमा, क्यों? मेध्य जो 40 ईयर्स में आता है, उसमें है बाल्य, वृद्धि, छबीर, मेधा। आप काउंट करो, हर 10 साल में हमारा शरीर में अगर 100 ईयर्स का है, हमारे बड़ी जीवायु शतेर भवेत। तैतरीय उपनिषद में कहा गया है कि एक इंसान का नॉर्मल एवरेज 100 ईयर्स, इसमें हर 10 ईयर्स में कुछ कम हो जाता है। क्या है? बाल्य, वृद्धि, छबीर, मेधा, 40। तो ये जो 4 करके निकल जाते हैं ना, उसी में से अगर स्टार्ट कर देते हैं, तो आपने जैसे मेध्य रसायन कहा, एंड दैट शुड बी अ प्रोजेक्ट आल्सो। बाल्य, वृद्धि, छबीर, मेधा, त्वक, दृष्टि, शुक्र, विक्रमौ, मनः कर्मेंद्रियश्चापि जीवितो हसेत। यानी टोटल 10 ईयर्स का माइनस हो जाता है, तो 100 ईयर्स तक आप जीवित रह सकते हैं। बहुत बढ़िया किया आपने, इतना सुंदर काम है। और अंत में आपने बोला कि जो है मैडम, कि जो हिडन ट्रेजर बीर्ज विपाक के लिए जो आपने कहा, ये एक न्यू डायमेंशन है। वी हैव टू अंडरस्टैंड इट अकॉर्डिंग टू आयुर्वेदिक कंसेप्ट एंड ऑल्सो बाय मिक्सिंग टू द मैडम। थैंक यू मैडम, ए लॉट ऑफ क्लाउड दीजिए, खूब जोर से तालियां ठोको। थैंक यू। अभी हम दूसरे जो वक्ता हैं उनको बुलाएंगे। प्रोफेसर अस्मिता वेले, पहले तालियों से तो स्वागत करो। इज करेंटली सर्विंग एज प्रोफेसर एंड हेड ऑफ आयुर्वेद फार्माकोलॉजी डिपार्टमेंट एट भारती विद्यापीठ डीम्ड यूनिवर्सिटी पुणे। Her expertise lies in Ayurveda-based drug development for asthma, cancer, and neuropsychology. बहुत ज़्यादा काम किए हैं आपने, वाह। And also she has successfully completed 3-year tenure Hungary as a chairperson and has been awarded honorary professorship at University of Debrecen, Hungary. एक तालियां और हो जाए। She has also sustainably contributed University Ayush. She was the expert, Ayurveda expert and co-rapporteur for the historic working group meeting for development of WHO benchmark documents on Panchakarma, Ayurveda practice and training. Thank you, ma'am. You are welcome.
जी मैम।
सिर्फ डाउन की है। ओके। हाइट, नमस्ते। At the outset, I would like to say my heartfelt thanks to Adaraniya Swami ji, Acharya ji, and the whole team of Patanjali University, PHRD, who have invited us here and who have given us a wonderful opportunity to share the work what we have been doing for past 3 decades. मैं प्राइमरीली मैं भारतीय विद्यापीठ में काम करती हूँ पिछले 30 साल से। जैसे ही अभी यहाँ पे बहुत सारे स्टूडेंट्स दिखाई दे रहे हैं आयुर्वेद के भी और बाकी ब्रांचेस से भी, तो जैसे ही पास आउट हुए भारतीय विद्यापीठ में चले गए, नया था, उभर रहा था, बहुत सारा स्कोप वहाँ पे मिला, तो भारतीय विद्यापीठ ने जो हमें काम करने की संधि दी, basically we were free to take up any ideas, and that's how our whole group has been successful today. And यहाँ पे जब आई हूँ तो कल से हमारे कॉलेज में पढ़े हुए एल्यूमनाई मिले, दिल्ली में भी बहुत है, यहाँ हरिद्वार में आजू बाजू में भी बहुत है, आके मिलते हैं तो बहुत अच्छा लगता है कि चलो हमारे विद्यापीठ ने बहुत अच्छा काम किया है। डॉक्टर कल्पना मैम ने जो स्टेज सेट किया आज कि उन लोगों ने, मतलब जो नॉन आयुर्वेदिक हैं, उन्होंने आयुर्वेद के कौन से कॉन्सेप्ट्स पे किस तरह से काम किया, ये जो अलग साइड, सो दे हैव प्रेजेंटेड अ डिफरेंट वे हाउ दे हैव वर्कड ऑन आयुर्वेदिक कॉन्सेप्ट्स। Yesterday we also heard a few presentations from other side of the table. Now coming to the side of Ayurveda, we also heard Dr. Parameshwar Arora ji telling us that yes, we can take up one aspect of Ayurvedic dinacharya, like water, when to drink, how to drink, how much to drink, when not to drink, which are the diseases where we need to as a doctor we need to take cognizance of water intake of our patient. तो इन दोनों को सामने रखते हुए I think I will present today neurocognition and rasayanas, how we did and what we did. In Ayurveda we always understand that health and well-being, they go hand in hand. They are two sides of one coin. We can never separate a coin, no? This side and that side. So either you are healthy or you are unhealthy or diseased. So either you are swastha or aswastha. In the scientific terms, we call it prakriti and vikruti. So either you be in your prakriti, be swastha, or you are aswastha and there is some vikruti which is underlying. Now these prakriti vikrutis, they both are physical as well as mental. When it comes to mental, there are two terms which we find: emotional health or mental health. So prasanna atma indriya manaha swastha iti abhidhiyate. That atma indriya mana complex talks about emotional health or maybe mental health. Mindfulness, consciousness, these are the terms which are going on in today's world, and all the Ayurvedic students must be aware about how people are looking at us. Once we know exactly Our science, I assume that we all are studying good, we are studying Samhita's text, but the gap that how we are going to apply it in the field, that needs to be filled, and which will be completely filled when we know what people want from us in the field. So we need to understand these terms which are being very regularly used on national and international platforms. What happens, a little bit of deviation, but I want to share this experience because it will go further in time. When I was there working in Hungary for 3 years, I was working in a medical school. That was not a Ayurveda institute or Ayurveda school. It was a medical school. University of Debrecen is a big university having 30 plus schools and departments. The medical college, School of Medicine, that was also very old. It is a university which is over 540 years old. And that medical school hosted Ayurveda department, Ayurveda chair, with an understanding that I will be able to contribute to the present day knowledge. So had I not been trained into this learning from both the sides, it would have been difficult for me. But fortunately, the mentors which she is mentioning, Dr. Kalpana madam mentioned, I also happened to train in that school for a while, some time, because I am primarily from Ayurveda. So Dr. Sharadini Dahanukar, Dr. Darshan Shankar, Dr. Narendra Bhatt, Dr. Bhushan Patwardhan, everybody has contributed in this understanding and growth. And let me tell you that knowledge coming from all the sources is always welcome. So unless and until you understand that and you try to look at your own science from the other person's lens, you won't be able to know yourself better. So with that, with this understanding, I started working there, and a professor of psychiatry department came to meet me. And then he said, what is there in Ayurveda for psychiatric diseases? Which drugs you give? What do you do? Do you do counseling or not? Then I had to explain that yes, we have worked on both the sides. There is a pharmacological approach and there is a non-pharmacological approach for cognitive health or brain health. So today we are not going into the details of the behavioral aspects, but on the disease aspect. Here comes the role of rasayanas. As my senior co-chair narrated the shlok very nicely, I have decoded that shlok in this table. That when you want to maintain health, when you go towards preventive and promotive health, then health needs rasayanas. When exactly? Are there certain time points where for a healthy person can we advise some drugs and which drugs and where? So here we get these decades of life. So we know the growth milestones, no? Adolescent, it's 0 is birth, and then toddler, and then child, and then adolescent age, and then adult, and then old. But Ayurveda doesn't do that. Ayurveda gives a more subtle classification. Ayurveda tells that from 0 to 10 is balya. So at the age of 10, everybody starts losing the childness in him. So the factors, subcutaneous fat goes down, the child starts looking lean, the long bones start growing up, and they don't know where to keep, how to use them. 10 to 20 is physical growth time. So at the age of 20, generally this factor should be taken into consideration. Similarly, at 30, there is start of loss of complexion. So at the time of 30, at the age of 30, you should be advising everyone to take care of complexion, certain internal as well as external applications. At the age of 40, there is loss of medha, knowledge retention power. At the age of 50, tone and luster of skin starts going down. At the age of 60, shukra or sexual vigor starts becoming less. At the age of 80, Vikram starts going down. So at all these, these are the decades where you lose one important factor of human life. And at this point, at the beginning of the loss, we should introduce the rasayan. Today's topic is rasayan, so I especially put this slide. We all know that, okay, this is there in Ayurveda. And lot of things are there, they are proven, we are practicing. There is Chitta Chatushtay when we talk about neurocognition. Antakaran Chatushtay or Chitta Chatushtay concept. Shira, Hruday, Basti. And these are three marmas. But in the Chitta Chatushtay, Buddhi, Man, Ahankar and Atma, 4 are considered. Now, how to research on them, what to do? That was the thing. And when we started, we said that there are anecdotal documents, but we do not have real evidence that, okay, I am an Ayurvedic practitioner, I have been analyzing these many patients based on my understanding, my proforma of Prakriti, my proforma of Vikruti, this is the Dhatu set, this is the dosha, this is the disease, Lakshana Samuchaya, and I am choosing these type of therapy modules, and then this is my, these are my results, and I have a data of 1,000 patients. I had to face these issues that you are so old, you don't know your origin, it's maybe 3,000, maybe 5,000, and still you can't give us data on 1,000 patients from such a big country where you are practicing regularly of a condition. We are not asking you about a drug. We ask you about a condition that yes, we have been treating this condition successfully. So that's where we have limited evidence and anecdotal documentation. On the other hand, when we started searching on brain and mind, I came across the systematic experimental documentation. I came across this approach of modern science. Some may like it, some may not, but this is an open acceptance that we don't know this. We know only this much, and we will find out how to know which we don't know. So that approach appealed me first. And therefore we started with studies on ghee for efficacy evidence. So cow ghee was the comparator. Dr. Kalpana ma'am has written a wonderful paper, she and her group, that the Ayurvedically prepared, that is the traditional cultured ghee, is better in all the qualities, and we all know that how ghee is used. So this ghee, if consumed every day, is giving us rasayana, jivana, tvacha, medhya, vrusha characteristics, then definitely that is going to work on patients as well. Then we have this Hingvadighruta, Vachadighruta, Brahmadighruta, Panchagavya, Jyotishmatyadi, Amalakyadi. All these are labeled under Unmad Apasmar Adhyay, Charak Chikitsa 9 and 10 chapters, as well as Ashtanga Hruday Bala Chikitsa. So we have studied on all these ghees which are there, which are listed here. But today I'll be presenting only one work, that too just as an example. So we put together this Hingvadighruta. This was not available in the market. We searched for, while putting together this Hingvadighruta and studying for it came in the Unmad Apasmar Adhyay. So we had to first ascertain the identity. The pharmacognosist came in, pharmacologist came in, pharmaceutics person from our pharmacy college. We all sat together, we brainstormed at how we should go about it. Then we prepared this ghee using this Hingu, Ferula, Narthex, Savarchal Lavan, Shunthi, you know that the standard drug, Zingiber officinalis, then Piper nigrum, Piper longum, cow urine. Again, this cow urine was collected, standardized, and then it was used. Cow ghee, traditionally Ayurvedically prepared, standardized, and then used. Before going into that, we also searched, because here our approach was from Ayurveda to modern. So we searched that which are the activities which are attributed to all these drugs. So Hingu is studied for memory enhancer, anticonvulsant. Shunthi is studied for antidepressant activity. Marich has been proven for anticonvulsant, antioxidant, sedative. Pipali, you know, piperine is a wonderful adjuvant. Then CNS stimulant activity Pipali shows as a whole. Savarchar Lavan supports this was another revelation to me. I read all the Ayurveda granthas, but I never knew that Savarchal Lavan supports resting of the body, sleep, and that is supported by a Lavan Gomutra, memory enhancer. And Gogruta, of course, memory enhancer, antidepressant, anticonvulsant. So when we studied that, okay, we already have a lot of data coming from scientists outside Ayurveda who have studied on all these activities. Now, what's the need for us to go ahead with activities? That's a very basic question. So we all agree upon now that if we have something to show clinically, we can replicate it in the lab, and then we can create more understanding. We can enrich the already existing knowledge with contemporary evidence. So here for Hingvadi Ghruta, all these activities we decided to carry out. Out of that, antipsychotic activity coupled with anticonvulsant activity, then anti-epileptic activity coupled with depressant activity, and nootropic activity were conducted in vivo. I'll present it in the next slides. And then antidepressant activity we conducted clinically. Yesterday's presentation, Dr. Bhattacharya ji has made my job very easy, so I have removed those slides because we also used elevated plasmase, we also used tail swim test and all other mice experiments. We did total 9 experiments for all these, for finding out all these activities, and for doing these experiments we worked with our Bharati Vidyapeeth Medical College Pharmacology Department. So I am a person of Rasashastra Bhaishajya Kalpana. And we have a team of our pharmacy people as well as our pharmacology department of medical college. Under Dr. Vijaya Pandit's guidance, we worked there and with those colleagues. So here the results, Hingvadi Ghruta showed antipsychotic effect when compared with standard drug haloperidol and olanzapine without the side effect of haloperidol, which is extrapyramidal shock, catalepsy. So HG is not better. We also came to know that HG is not better, but it is effective. Then this Hingvadi Ghruta exhibited anti-epileptic effect less than the standard drug. Because standard drug olanzapine, the standard drug phenytoin sodium gave 100% result. This Hingvadi Ghruta gave 37.5% result, but that drug impaired memory, and this does not impair memory. So there is a window. Hingvadi Ghruta showed antidepressant effect which is comparable to imiprimine hydrochloride, and it also improves memory. After that, when we did pilot randomized control trial in 12 patients in mild to moderate, suffering from mild to moderate depression, in this study, we gave this Hingvadi Ghruta as adjuvant to one group for 60 days, and they showed significant results on the Hamilton rating scale. So, starting from the screening of what people have done, then formulating our drug, standardizing our drug, using our drug with animal models to the precision, taking help of pharmacologists, and then conducting a trial by completing all the formalities of randomized control trial like the ethics committee permission and registration at CTRI and all. Here are our learnings. What we learned, first thing, now when people of Ayurveda are looking at this presentation and other side, for both of them, I'm putting this, that can the modern drugs be replaced? I'm not going to answer these questions. The answers we all will find out together. Can the Ayurvedic drugs be co-administered? Can the time and usage of both the drugs be complemented? So each of these questions should need some actual field research before claiming some activities, or before saying that, okay, our drugs are good, our drugs are very effective, or modern drugs have side effects, they do not work. Nothing like that. Let us come to a ground level that, okay, my patient is important, and what the patient needs, if both the sciences can deliver it for the patient, then it is required. So generalized perceptions after this study where comparator is cow ghee. In all our studies, I'm not showing the graphs and all because of the time constraint. So comparator cow ghee is food. It is effective. So every day, every person must have cow ghee. Now when this is effective, whether medicated ghee is better than cow ghee or it is similar. If it is equivalent, then there is no point in, trying all of this. You know how long it takes to make a medicated ghee. You have to identify everything, then make juices, decoctions, boil it, keep it overnight, again boil it. So the whole process goes more than, for a week, and in some cases more than a month. Is it worth it? So after this study, we are 100% sure, and I want to make all Ayurvedic physicians assure that we have a report that medicated ghee is 100 times better than cow ghee, because it is better, it shows better results, and therefore it should be used for the expected rasayan purpose or disease purpose. What should be the dose of the medicated ghee? We had done, along with these big studies, we have studied on a lot of things, bhasmas also, but here for this particular study, we did a small survey of Ayurvedic Vaidyas. We are blessed that Pune has a very huge, long tradition of Vaidyas, and they practice very traditionally. So we sent our students with questionnaires, and there we could get that the textual dose is about 40 grams per day. But in practice, Vaidyas generally prescribe 20-25 grams. This is because of the patient compliance. Most of the patients, they do not, because of this, publicity that ghee is fat, reduce fat in your diet, this and that, and people fall prey to that. It's not their fault also. So what they did, ghee, no. Some people didn't like the taste of the ghee. So the dose generally goes between 20 to 25 grams, and our results are matching with the text dose, that is about 1 pala 40 to 42 grams. We need to refine the dosage form and mode of drug administration. So if we want to see the result, then definitely there needs to be a drug development approach adopted for these effective but problem with palatability doses. After these studies, all these studies, we could come to a conclusion. There is clearly a grouping of herbs from Ayurvedic viewpoint. The first group of herb consists of Haritaki, Vacha, Shankhapushpi, Pippali, Jirak, Jyotishmati, Rasona. They are predominant in Katu Rasa, Katu Vipak and Ushnavirya. They work on Vata Kaphaja type of disorders. So even if it is Unmada Pasmar, there is a classification we know, Vata Pradhanya, Kapha Pradhanya. If we want to use Ghritas having these herbs, then that will go for this type of disease. Disease manifestation, Vata Pradhan disease in a, now if we go into the intricacies, Vata Pradhan disease in a Kapha Pradhan person and Vata Pradhan disease in a Vata Pradhan person, which is difficult to treat? Which is difficult to treat? Ayurvedic students here? Vata Pradhan disease in a Kapha Pradhan person, difficult to treat. So this group of herbs will work there, and the second group of herbs consisting of Brahmi, Yashti, Madhu, Shatavari, Dadima, Jata, Mausi, having predominant Kashaya, Madhura, Rash, Shita, Virya, it will work on Pitta Pradhan diseases. So a group of these neurological diseases. So there were, out of all these studies, I have tried to put together here a few thoughts also. We should be going with the preventive and promotive approach. The easiest approach is through food supplements, daily consumption guidelines in alignment with Prakriti. Definitely that can be done, but when I say that can be done, we should be able to get data from that. So even if here we do advise certain pathyas based on Prakriti, certain drugs, there has to be certain mechanism by which we will know what they have done, how they have adhered to the diet or restrictions what you have prescribed, and what is the real outcome of following all that. Similarly, when we talk about Ayurveda, everybody wants that Ayurveda should be propagated out of India, but basically we need to think about our own country. We are very huge population. We have a lot of consumers, and therefore there is a lot of chances, there are a lot of chances to get data from our own people for our own people. So what I learned is a small but impactful study is the first thing which we require, and then we can go for scale-up operations. This is where Ayurvedic process of cognition leads us. Atma manasa sanyujyate manaha indriyena indriyam arthena tato jnanam. So if you can see the blue arrows at each of this point, this is a point of intervention of a food or a drug for each of these activity. So there can be a problem in desire, there can be a problem in stimulation, there can be a problem at indriya level, there can be a problem of process level, processing of the information. So there is indriya buddhi, there is medha, there is knowledge acquisition process. At each of this point, there can be some drug or some non-pharmacological approach coming from Ayurveda, which the new age student should try to look for research leads and then come up with different models. And here are the questions that need answers. Vaidyas, they practice sincerely, genuinely, they examine the patients, they analyze, and they treat the patients as per the standard Ayurvedic templates. It's there in their head, but they do not record. There are lots of patients, their question is genuine. If I'm seeing 80 patients a day, where there is time for me to, record everything and then go back and then dig into the data and come up with something. So here universities need to strengthen the hands of practicing Vaidyas. Universities can prepare certain, we can collaborate. We can prepare certain templates, we can prepare Excel sheets, data storing mechanism, and students can go work with those Vaidyas for those particular diseases, and then we can sit at the back end and analyze and come up with certain evidences. Evidences show change in biochemical entities like cholesterol. You have seen Kalpana ma'am's presentation, the effect on different kinds of genes with, you have seen it, I don't need to repeat it. But as a Vaidya, what should I do? Should these be used from the other approach to reduce increased cholesterol, advise Rason, or procedure of Virechan? Should I use that way? Or should I continue my own practice? Like I have been trained and treated with certain, documentation practices. And if after these studies, people start using Rasona, people start using Arjuna as cardiotonic, people start using Rasona as anti-cholesterol. Should I stop them being a Vaidya? Because this comes from Ayurveda, the knowledge base, the knowledge lead. What should we do? I think we all need to sit together and deliberate upon these questions, because there is no one answer to this. The only thing I feel after all these years is contribution by way of generating data, putting it properly in front of the world is essential. And since I am associated with this international platform also, I know that about 77 countries Ayurveda is existing. Different therapists, some doctors, some nutritionists, they are practicing and using Ayurveda for their patients. Where is the data? They are using it, but there is nothing. So we have to think about this type of approach, and I don't think that any other platform rather than the Patanjali University is right now able to do that. So this big task should be kept ahead. So in neurocognition and rasayanas, at least on India based, I feel that there should be prospective studies on healthy volunteers by adhering to standard protocol, meticulous data collection and conforming to the ethical approach we can do this, and documentation of real-time data. So there can be prospective planned studies as well as documentation of real-time data can be done. Here is my team. You can see the whole Bharati Vidyapit, my students, pharmacies which support us generally, my principal. You can see here in the middle Dr. Vijaya Pandit ma'am, who is with us for past 20 years. the initial meetings were very heated. She wanted to understand what is Ayurveda. I wanted to understand how do she looks at Ayurveda from the perspective of proteins, fats, carbohydrates and different drug moieties and molecules. But we have come up with wonderful projects, and I hope someday some of you would be able to visit us and see how we work together. With this, I thank you for the patient hearing.
थैंक यू मैडम, आपने बहुत ही अच्छा काम किया है, इसमें कोई शक नहीं है। रसायन के बारे में जो आपने बताया, और दैट इज़ अकॉर्डिंग टू 10 years ऑफ डिग्रेडेशन द बॉडी, सो दैट इज़ मोर इम्पोर्टेंट। और वो भी इससे पहले जो मैडम ने बताई थी, आई थिंक दैट इज़ मोर हेल्पफुल फॉर द ऑल दिस नेक्स्ट जनरेशन। अच्छा, सबसे जो आपने बताया हिंग HG, जो हिंग घृत, तो इस तो ठीक है, हम मान लीजिए घर में बनाते हैं, बट प्रॉब्लम है कि हमें कहाँ ये देसी घी मिलेगा। मैं जब लखनऊ में डायरेक्टर था तो वहाँ पे मैंने मार्केट सर्वे करके, हमारे कॉलीग यहाँ बैठे हैं वो भी जानते हैं, डॉक्टर पाठक भी हैं, ही वाज आल्सो इन CCRS। मैंने मार्केट्स डिफरेंट कंपनीज के कुछ घी के सैंपल लाए और उसको लैब में ले गए, वहाँ पर सीमैप भी है और ये भी है। तो देखा गया इसमें से स्टैंडर्ड को भी कोई नहीं मीट करता है। तो हाउ कैन वी प्रिपेयर हिंग घृत, और देशी ब्रैकेट में अब लिखा है देसी। एनीवे, हर एक जगह तो गाय हम रख नहीं सकते हैं अपने घर में। आजकल देखे हैं जो मार्केट में चल रहा है एक कमर्शियल एटीट्यूड्स, किसी तरह का उसको जो है इंजेक्शन दे के ज़्यादा दूध निकाल के उसको घी बनाना। सो ऑल द थिंग्स आर देयर, but the parameters you have adopted is definitely okay and as per the research guidelines, and it will help also to our next researchers. And the question आपने लास्ट को किया कि not to be answered, वो सवाल आपने सही में पूछा कि अभी क्या एलोपैथी को हम रिप्लेस कर सकते हैं? मेरे विचार से नो, क्योंकि अभी भी देखिए जेब में अगर आप जो BP पेशेंट है वो ज़रूर उनको जो है टेलमासेटिन्स होगा, शुगर का पेशेंट हो तो उसका जेब में भी होगा। लेकिन अगर अभी अभी इमीडिएट किसको वोमिटिंग हो रहा है तो एल्बुमिन के टैबलेट ज़रूरत पड़ेगी, और भी दस्त हो गया तो उसमें से नॉर्फ्लॉक्सासिलिन की ज़रूरत पड़ेगी। तो इसको हम रिप्लेस नहीं कर सकते हैं, बट इट कैन बी केप्ट एज आवर इमरजेंसी नीड। और बाकी जो सवाल रहा कि जो कैम्प में हमारे आयुर्वेद आया हुआ है, इससे तो बचना या निकलना बहुत मुश्किल है। खैर, आप लोग जैसा साइंटिस्ट अगर रहेंगे और दिग्दर्शन देंगे तो हमें लगता है आने वाले दिनों में कि हम इस कैम्प से निकल पाएंगे। थैंक यू मैडम, थैंक यू द ऑडियंस।
Thank you, sir.
Hello. Our next presentations are going to be online. So by the time they get set, when they give me the signal, I will introduce the speaker. Please, let's have some conversation.
Madam,
Welcome.
Both the madams in the morning have raised very important questions, and you also said very correctly, sir, that we need before to the traditional allopathic medicine. Once you show that, then you can go for a larger study, multi-center study. So we need large trials.
Dr. Sir, a very welcome suggestion, and maybe we can work out some modality at the end of this session. We can converse it. Now, Dr. Prithviraj, can you hear me? Hello, Dr. Prithviraj, can you hear me?
Yes, I do hear you.
Wonderful. So, most welcome to be here. We are in Haridwar, welcome you. Let me introduce you. He is founder, director of the Microbiome Research Laboratory as a part of Department of Immunology at Dallas, Texas, USA. He has received several awards, And they are very noticeable in the sense that the Hartwell Foundation Biomedical Award and Synergy Grant Medical Award, so you can see that those awards are in the areas that we are now today interested. He has received the Young Scientist Award and several others. He has been involved with the pediatric infectious diseases and so on, and also immunology activities, immunological research activities. He has been editor and reviewer of the many, as a part of many journals. So let us welcome Dr. Prithviraj. We are eager to know your views and experiences. Welcome.
Thank you, Chairpersons. Do you hear me okay?
Yeah.
All right, great, thank you. So good morning, India. This is kind of midnight for us in Dallas, Texas. So, and I really, it's a great pleasure and great honor to be part of this great event. I really want to thank you, Swami Ramdev ji and his entire team, Dr. Ved Priyaria, Dr. Aswini Kumar, and Dr. Gautam for inviting me for this seminar. And I really, learned a lot from the previous speakers as they put kind of, primed all the audience for what I'm going to talk about, mostly the genomics technology. So let me check on this. My slides are moving. So I thought, I certainly not working on the Ayurvedic genomics currently, but the technologies that right now we use in our lab, we do like human genomics research, and as you mentioned, we study pediatric populations and their susceptibility to infectious diseases. So there are a few things I put on this first slide, just to start with what we know about the herbs is kind of, what previous speakers alluded to for medicinal plants that have therapeutic potentials. And we know that for like a long history of humans, they can treat ailments. And Ayurveda is one of the ancient, I think, universal natural healthcare systems. It's not only in India, but I think if you go to Europe, you go to Western, further Western in south of the America, we do see, people practicing Ayurveda. And we know that these, natural products that we have right now in practice, to mention few of them, that has come from various kind of medicinal plants, aspirin and atropines, and we use them currently in different medicines. They all come from these herbs that, people kind of, practice for various kind of, health issues around the world. But what I think, we do not know precisely, as previous speakers also mentioned, and I appreciate like these efforts are being done in various great institutions in India. Now, what we don't know precisely is the mechanism of action of these, when we talk about a compound that is coming from the plant system. What is the molecular biology? What is the mechanism? How does it work? Does it work on the B cells? It works on T cells? It works on your myeloid lineage cells? Or does it work on neurons? So that kind of precision is something that we want to learn. We want to learn what is the optimal concentration of the compounds that gives the most beneficial or therapeutic potential. We want to understand how they interact with various, human systems. Given a medicine, does it interact with the livers? Does it goes to the kidneys? So all that kind of precision pathways. We also want to understand like the species level of heterogeneity in the pharmaceutical potentials. And I think, for wide variety of the herbs, we still want to understand what are their clinical applications. So as the Dr. Joshi and the other speaker mentioned, so the current practice right now, when we go to any kind of medicinal pathology treatment using like the traditional medicine strategies to really, give the general medicine and then see who get benefits and who does not. But what we want to do is now trying to get these efforts to really understand the genotype and match a right therapy to the right genotype so that we can get best benefits. So we are right now, I think, struggling around like stratified medicine, where we are able to identify groups of people like based on ethnicity, based on genders, based on the genotype compositions, we are able to understand what kind of immunoregulatory therapies, for example, in case of cancers, are most beneficial. But where we want to go ultimately is to really go to the personalized medicine, where we want to understand like what medicine is really perfect for a given person. So what underlies this whole understanding and the objective is to understand the central dogma. Like we want to understand the molecular biology, be it a human cell, be it a plant cell. We know there is a DNA that makes RNA, gives the instructions to make the protein, and then protein express. That underlies the whole mechanism, and we want to understand it to cell to understand this mechanism. I put this slide to really attract the attention of, like, there are several students I see. So what we want to understand, like we are looking at these carrots here. So we know that there is a this part of the carrot, you see the whole green, that is shared across these different species of the carrot, but you see the other body part of the carrot is really species specific texture of the color you can see. And qualitatively also, if you look at the nutrients components in these different types of carrots, that you would also see that they're heterogeneous, and there underlies a genetic instructions that applies, that is transferred to production of these different compositions. And that is differing between all these different species. But the part of the DNA that is encoding for this, green part, that is perhaps shared across these different species. So this is where we want to really understand how genetically these traits are regulated in different plants, and then we can precisely get to their functionality. I believe that plants are the great model system to really learn how these regulation of the genetic material to transform into the proteins, and to understand the efficacy of these compounds can be done greatly in the plants, as evidenced by these two major discoveries done in the past. Like Gregor Mendel used the green peas to really give us the fundamental laws of inheritance, how genetic traits goes from mom, dads to the next generations. And Barbara McClintock in New York, where she studies these maize, as you can see, she showed that you get some DNAs from your parents, and then there are genes that jump around. They don't stay there for all over. They keep jumping as there is a switching of the traits can happen, and she demonstrated this in these maize plants. So we still believe that this is true for the other DNAs in the other plant system, where you can see various new characters are developing over time. So currently there are several genomics techniques that, for example, we are using in human population studies, and these are the tools and techniques that can similarly be applied to the all kind of the plant system or in the herbal genomics. For example, you can take the DNA sequence information by doing the deep genome-wide sequencing of the given kind of herbal plants. You can do genome-wide association studies, or GWAS, where you can look for the SNPs or the mutations that associate with a particular type of trait. For example, the yield, the particular qualitativeness of a particular compound, and you can do various bioinformatics analysis to really figure out what codes for what kind of traits. You can do DNA marker kind of technology development. If you want to barcode a species, you want to do their phylogenetic analysis, you want to look at what kind of species have a shared traits, and you can do this more precisely on the functional side by doing precise transcriptomic analysis, where you can go for whole transcriptome analysis, sequence their RNAs, not only the message RNA, but also look at various different regulatory RNA species that are present in a specific species, and how they kind of regulate the transcription and gene expression. Further efforts right now as being done in case of the human genetics and in developing new gene therapies for various human diseases, like mutagenesis, where you can artificially induce a mutation. Either you silence a gene or you can activate a gene. Similar studies can be done in the plant system, where you can knock in and knock out a gene, and you can look for how the traits, how the particular compound's quality changes in a particular species when you kind of do a modification in a particular gene or a SNP. And for particular tools that are being currently used in the human genetics and human genomics right now are the CRISPR-Cas technology, which really allows you to very precisely go to a particular interval in the genome and do this intervention to really look for the changes. This is one example where you can apply DNA barcoding, and it is being done right now in many of the herbal genomics labs in Europe and in China. I guess there are several groups working on where the idea is you start with a specimen. For example, you pick this leaf and you can, extract DNA, you kind of sequence the DNA and you identify a unique phylogenetic signature that belongs to that particular species, which you can assign and use as a barcode in the library of herbals, medicines to be used in the future. So that gives a precise taxonomic identification based on the DNA barcoding system for that particular species to annotate for future research studies. You can also look for different compounds that a particular species is making. For example, we are using now these methods to really understand what kind of metabolites are being produced from a particular plant system, or for example, you take ajwain, for example, you grind it, you do the mass spec, and you can look for what kind of molecular compounds are coming from the ajwain, and you can further characterize the molecular basis of a particular quality that a particular herbal product or compounds kind of exhibit. So I'm going to show you some of the research that is ongoing in our lab, and we are interested in really understanding how, the herbal products, for example, the vitamins and the other nutrients that we get from various kind of, the vegetable or diets that we eat on a daily basis, how it interacts with our microbiota in the gut and oral cavity, for example, how we really derive these dietary fibers and short chain fatty acids, how they are being absorbed into our intestine, and how they really kind of give us those beneficial effects to really get the immunity against various kind of infectious diseases, autoimmune diseases, cancer, etc. And when we're talking about the human microbiome, we know that, if you look at across the whole human body, we are loaded with the trillions of microbial organisms. They are like in the oral cavity, they are present in your gut with a wide variety of diversity. They're all over your skin surfaces. And what we are talking about archaea, fungi, bacteria, proteobacteria, all different variety of species that kind of, are commensals. They're living with us for millions of years. They're evolving with us. So whenever we eat something, when we eat a plant, when we eat a non-plant products, that really directly interacts with the microbiota in our gut. And then microbiota kind of, help us really draw those nutrients, absorb those nutrients from these compounds. So we are interested in really understanding these mechanisms, at the molecular level. So the idea is we know that in the microbiota and in how it interacts with the host to really put us in a homeostasis position to really get away from the pathology. So the idea is, I'm showing you one illustration in the panel you see here, that these are a single layer of epithelial cell in our intestine, and you see that there are a lot of different kind of microbial cells like Clostridia, Fragilis species. And what we need when we are in healthy state, we need high microbial diversity in the gut that keeps the anti-inflammatory species on, and there is no translocation. These microbes stay on this lumen side of the cavity, and these cells are secreting this antimicrobial proteins and different antibodies from the single cell epithelial layer in our intestine, and keeping these organisms at a distance from us. And this, what happens on the other side of the immune system, when you have this distance maintained with our microbiota in the gut, you have all kind of, tolerance to the commensal, homeostatic effector functions are on, and we are able to regulate the immune responses. And this is what we call a healthy state, when T regulatory functions are up. On the other hand, when the microbial diversity is lost, when we have a certain kind of microbes get more expanded, we get only pathogenic bacteria expanded in our gut, and the good ones are kind of lost. You see that what happens now, we are not secreting that much of mucus, we are not secreting those or antimicrobial proteins into the lumen, and that sets the stage for all B cell pathology, Th17 upregulated T regulatory responses are down, and this is what we call inflammation, the pathological state. So we want to understand what happens when we are taking these all kind of, horrible things into our gut, how they really impact all these interactions inside the human intestine. And why we want to do this, why we do these experiments? We want to do this because we know that in a healthy state, you can look at the microbiota in your gut by doing the high throughput DNA sequencing. You can determine, for example, I'm showing you one example of there are 4 different individuals, C1, C2, C3, C4, and you see in their gut, and different color represent different type of bacteria present in your intestine. You see that all these 4 individuals have a different composition of their gut microbiota. Some people have more of the blue colored bacteria, which represent firmicutes, and other individual C1 has a lot of proteobacteria in their gut. So how that composition then, really make us like more susceptible to disease, more protective from infectious disease in the intestine. That's what we want to learn. And once we know that what's going wrong for example, in the cancerous state or in the autoimmune state, we can bring in diet, the right diet. We can bring in probiotics. We can bring in prebiotics and antibiotics to really restore the normal state of the microbiota in the gut, to treat the disease by modulating your microbial compositions in the intestine. And here is one discovery that we actually made in 2021. We all know, like in India, हम शुरू से जब बच्चे से छोटे से बोलते हैं carrot खाओ बहुत अच्छा है, right? We all eat carrots, and we know that carrots are, really rich source of vitamin A, and vitamin A is crucial to fight the infectious disease in the intestine especially. If you really knock out the vitamin A gene and make somebody really deficient for vitamin A, then that person will develop a lot of salmonella and other intestinal infections rapidly, and there will be a lot of pathology and disease going on. So this was a study that we conducted, and I'm just showing here illustration just for the purpose of this audience. I'm not going detail into the mouse studies that we did, but the idea is I'm showing you, this is all the story about the intestine. You see, as I showed you in previous slide, there is a lot of microbiota. There are different kind of bugs that stays in our intestine. They're all friendly with us most of the time. When you eat vitamin A, you directly don't eat vitamin A, you eat the carrots, and carrots has retinol or the product that comes from the vitamin A is converted into retinol in the epithelial cells. So I'm showing you illustration of the intestinal epithelial cell. So we found out like it is converted into retinol, and there is a specific protein which is called serum amyloid. So serum amyloid is the one that carry now the vitamin A that is coming from carrots to the intestinal myeloid cells such as DCs, macrophages, and then it is converted into retinoic acid, because retinoic acid is what our T cells and B cells need to make antibodies to fight the infection. So B cells are the antibody producing cells. So what happened in a general infectious disease, whenever our body encounter any pathogen, our DCs or the macrophages or antigen presenting cells are the professional cells. This is our security forces that tells the central government, they tell the state government, like they talk to T cells that we have seen something like a foreigner, we have seen somebody that we don't know about. And then T cells talk to B cells that, hey, do you have any information of this kind of pathogen? Have you made antibodies earlier about this? No, I haven't. So then B cells start making specific antibodies to keep, for example, salmonella kind of enteric pathogens away from the intestine. So now you think about when we goes down with the carrots, we goes down with the vitamin A, you are going down with your adaptive immunity, which means CD4 activity, your T cell activity, your B cell activity, and you are making less of these antibodies, or this is basically IgA types, where then you become more vulnerable to infectious disease in the intestine. So we eat carrot, but how it is actually molecularly and mechanistically transferred to the B and T cells, which are looking for this vitamin A or the retinoic acid to really perform their role in the intestine. These are the mechanism for any kind of herbs. I think in the next future years of this investigation, in case of microbiota and herbal genomics, transcriptomics, we are going to work on. We had a student who was interested in understanding that we know that from the, from our childhood, whenever we have anything wrong going on in the stomach, somebody have some flatulence, they say, okay, take some ajwain seeds, right? So we always wonder, right? So this student was interested, and it was like a summer student for a few weeks. And what she did, she took some, we had some volunteers, and they were, looking for this interesting projects to participate in, and we hypothesized that perhaps ajwain seeds relieve flatulence and provide any medicinal health benefits by modulating the oral or the gut microbiota directly. So this is our hypothesis. We don't know whether ajwain directly talk to microbiota, or does it, do something to the host side, and the host side then transcriptionally activate some pathways, and that relieves the flatulence. So the idea of the experiment is something like this: you're gonna take the ajwain seed early morning without eating any, before you eat anything, and then you go into the 30 minutes wait time, and then you kind of get another saliva samples to do it in the oral cavity, and then you wait another 60 minutes, and then you take another sample, and we take all these saliva samples, we do DNA RNA extractions, we do microbiome sequencing, and then you look for what you are finding. So here are some results. So we had like these kind of 6 participants I'm showing you in this panel, and we saw one bacteria which is a common normal flora, a member in the oral cavity. You see this is called Rothia, and in the control experiment, means before eating the ajwain, this was the amount of Rothia detected in the oral cavity, and you'll see after treatment represent when the person has already eaten the ajwain seeds and wait for 30 minutes, and then you see that there is a very clear upregulation, like the enhancement of the Rothia particular OTUs. That means that perhaps the ajwain is promoting the expansion of Rothia in the oral cavity. On the other hand, this is not true for bacilli. You do have bacilli in the oral cavity, but when you take the ajwain in, and you don't see on the same participants, you see there is no such correlation. There is rather kind of decrease in the abundance of the bacilli in the oral cavities. So I think these are some example experiments that we need to kind of precisely do in the animal models. We want to do them in case of the human participants on various the herbal drugs or herbal products that we have in our kitchen, to really kind of get into the mechanism of what we are practicing since childhood, and we know that it really benefits. But I think to really understand the mechanism so that we can apply it and we can modulate it to get the maximum benefits, we have to get to the real mechanism. So here is a kind of summary. What we are learning with the herbal genomics, and I think there are a lot of microbiome research right now going on to understand how this Ayurveda medicine is really enabling or really able to draw the most of the benefits that we get from these medicinal herbs. This research is kind of really facilitating the characterization of metabolic pathways in medicinal plants, enabling the discovery of novel species and the metabolites associated with them. Also, identification of novel enzymes that involved in pharmaceutical compound biosynthesis. And I was reading a few papers where they are really able to genotype, using a whole genome sequencing methods, really genotyping the pathogen-resistant strains, and also looking at if their yield is better than the other genotypes. Also, my personal interest is really understanding like how herbal compounds, for example, such as carrots and so many other things that we know that really provide benefits, how they impact the microbiota and the gut, and perhaps microbiota is the middleman really helping us really fetch those all good benefits from these compounds. So this illustration is something that I was thinking about. We as a human race. We came from these, rich, environment of, vegetables, forest that gives us life. And we went into the cities, which were kind of, darker side. We wanted to go there, but we feel like we still have to learn a lot, and we need to make this U-turn to really learn how to be healthy, I think, in the future. So with that, I want to stop here for the 30 minutes talk. I just put a few slides here, and I want to really thank you, my team here at UT Southwestern, who is wonderful and doing all these interesting experiments with me. I want to thank again the organizers, chairpersons, for chairing this session and allowing me to present and share some of the thoughts. And it's really great pleasure. I want to especially mention Dr. Ajay Gautam, and I have been like colleague for several years in India, and it's a great honor to be participating in this conference and listening to great science, and Ayurvedic, genomics that is going on in India. So I see a great future there. Thank you so much.
Big clapping for Dr. Prithviraj. Thank you, Dr. Prithviraj. And you have nicely presented your paper, which has having a lot of information related to our Ayurvedic concepts. And exclusively you have told the genomics and how the drug works in the body. And according to Ayurveda, kinchid rasena karute karmam birjena chaparam prabhavena chandrabhyanang vividhagati. Drug has got some multi-dimensional actions. And you have also told very nicely through your modern research. A lot of thanks again, and it will be help to our students and our next researchers. And thanks. 1 minute, please, sir.
Hello. Dr. Raj, thank you very much for a fantastic presentation. It's like very nice and good to see that there is a lot of thoughts going on looking on herbal medicines and how they can modify. The data that you showed about ajwain seeds and how they're modifying the microbiota is fantastic. So very, very nice degree of congratulations to you. But I have just one query. You have measured the oral microbiota status while giving the ajwain seeds. Any data do you have for the gut, deep gut microbiota? Because that's where ajwain might be playing a significant role. I understand it might become technically difficult to measure it, but has someone really tried that?
So, no, I think that's a great question, and I'm glad you asked it. So, here is the answer. So this, we know that as you know, that really doing this experiment on the stool sample in the gut, it's difficult, right? How you would say that it is because of ajwain that you took, right? You ate your dinner, right?
Right.
It could be your dinner's components, it could be something else, right? But what question the student asked, I mean, she was only for 8 weeks and it was a summer student's experiment. So our hypothesis is that if ajwain really interact with a particular bacteria, and since gut and oral cavity do share 60 to 70% of microbiota on an average, we will be able to find a particular bacterium or bacteria, at an order level or family level that is showing interaction, and perhaps similar interaction we can then, see when we do the stool experiment, I mean, in the gut intestine. So to really, I agree with you, the real experiment will be to see how does it impact in the gut microbiota. But to technically see, is there a bacteria that both the niches share and ajwain has a direct impact. So we were surprised to see that within these 6-7 participants, we see a significant enrichment of the rotia specifically, with these 30 minutes, and we are also looking at some transcription, like we did some RNA sequencing on some of these participants where we see very high. So, but I agree with you, yeah, the real experiment in the gut would need more, we need to really set that up. How do you sample for that? And that is technically a little challenging, but we will try to do that first in the mouse model system. Because we have mouse who don't have any microbiota, we call them germ-free animals. So we're gonna give the ajwain to germ-free animals and look what happens, right? Then we can bring in rothia, only rothia in the germ-free animal, and then see what happens. And then you bring the bacilli in the germ-free animal and then see what happens. I think these experiments then shed some more light on it. Yeah, that's a great question. Thank you.
That's a good idea that you talked about. One very quick thing could be done to do in vitro microbiology and grow Ruthia there and then give the extract of ajwain or ajwain powder as such and see what's the level of growth really happens there. So if you're getting a better colonies or getting a better growth in a time dependent or concentration dependent manner, I think that might help to go for the in vivo model that you just talked about. But thank you very much. It was a nice presentation. We appreciate that very much.
Thank you.
Thank you,
Appreciate
Dr.
that.
Raj. And now we will move to next, Bhavana Parashar. Dr. Bhavana Parashar is online.
Yeah, I just wanted to confirm if my slide is visible.
Okay, ma'am, visible. Just 1 minute for your introduction, ma'am. Number one, that is Dr. Bhavana Parashar pioneered the development of the new field of research, Ayurgenomics at CSIR IGIB. Her research integrates disciplines of Ayurveda, modern biology, computational methods, and genomics for developing a aid to novel discoveries of positive markers and therapeutic repurposing of Ayurveda interventions for modern diseases. Ayurgenomics as one of the 80 success stories identified by CSIR on the occasion of CSIR 80 years celebration and during Azadi Ka Amrit Mahotsav. She has awarded several awards and is the member of several reputed associations. Welcome, ma'am. You can also come to the point. Thank you, ma'am.
Thank you. Thank you very much for the nice introduction. And at the outset, I would like to thank Swami Ramdev ji, Acharya Balakrishna ji, and his wonderful team for putting together this international conference and providing me an opportunity to share some of our work. In this, I'll try to remain confined within my time. And since a lot of discussion has happened already over Ayurgenomics and how people have done several kind of work, seminal works in this area, I would like to connect this Ayurgenomics with the herbal genomics, as well as try to also allude to how it leads to the translational medicine, and we narrate only one story out of the many things that we have done so far. So just to put you back in the context where earlier speakers also mentioned about the inter-individual variability in health and disease transitions, we all understand everybody, although healthy, someday or the other might actually experience some disease. But who will experience what disease and in what severity, and what kind of medication would be suitable even if people are suffering from same disease, is a big question right now faced by even modern medicine. So definitely there is a clear need for predictive approaches and personalized approaches for effective medication. Now, where does this brings us to the question, how is modern science trying to, or modern genomics trying to address this? Modern genomics is trying to look for the genetic variations that are shaping the variability, and there are several initiatives world over where human genetic variations have been catalogued. Not only cataloging, they have also moved on to understand their relationship with the disease as well as with the clinical phenotypes in different approaches like GWAS and FIVAS, in order to identify markers that will tell who will respond in what way and who will be more susceptible to what kind of disorders. There are more newer approaches are also coming, like that of extreme phenotype approach, where you divide the population into with respect to one single phenotype. For example, having moles, too many moles versus not having any moles, having some kind of a BMI very high versus low. So one feature people are taking and doing the extreme phenotype based approach to identify molecular signatures underlying them. Where does Ayurveda come here? Ayurveda, we know it has a 3-sutra Ayurveda framework which has Hetu, Lakshana and Aushadha described for both healthy as well as diseased. So these things are actually talking to each other in a manner that they can actually provide for a translational medicine framework where one can connect Lakshana to Hetu, one can connect Hetu to Aushadha. So the Aushadha is also given to address the Hetu, keeping in mind the present features that are manifested in diseases. And all this approach is of a translational approach, but with a personalized, because they say it is for the healthy and disease. It's not for the health or the disease. So that tells that there's a clear clinical application for predictive, preventive, personalized and promotive health. But at the same time, in the research, the same can be systematically deciphered, explored for making newer discoveries of biomarkers, novel therapies of drugs through understanding their mechanism, and exploring the system biological networks which can provide us innovative scientific insights. So with the aim of restoration and preservation and enhancement of health, Ayurveda has described this 3-sutra and how we went about exploring these 3-sutra in systematic manner. I'll just take you through in a while. So when we say 3-sutra Ayurveda, external environment, internal environment, clinical manifestations, medicines, and they all being connected to each other. How is it that they are, what is the medium of expression or what is the language through which they are cross-talking? That is something very, very unique. And although each of these aspects have got different parameters to understand them, for example, in case of diet and environment, we have ras, gun, virya, vipak. In case of Aushadha also we have those things. In case of Lakshana, we have got many kinds of physical dhatu Lakshana, dosh Lakshana, etc. described. But they all crosstalk majorly, as a first line of crosstalk is through Tridosha, which is called as common organizing principle, which is Vat, Pit and Kapha. These 3 doshas, when they are in the homeostatic condition, we call it as healthy or it is Prakriti. The same when they get in response to environment or lifestyle or diet or because of several reasons, if they go perturbed, then it causes diseases. And the goal of therapy is to bring those doshas back into the same homeostatic stress, and that is the goal of therapy. So that is how we have this common organizing principle of Tridosha. The same has been depicted over here in the form of pictorial presentation. If the yellow triangle is what we are born with, which is a Prakriti dosha, the red triangle is the one which keeps on fluctuating in response to environment, day-night cycle, season, age, and several other aspects. But so long as that fluctuation remains within this gray aspect of the adaptive space, we still enjoy health. But the moment it gets into it crosses that adaptive space and it enters into the disease zone, disease is there. And the goal of treatment is to measure, first of all, how much is the deviation from the normalcy and accordingly act. So they say, dosh pramananurupo hi beshaj pramana vikalpo and bala pramana visheshapeksho bhavati. So which means that one measures how much is the dosha praman, accordingly gives aushadha. So this is precision medicine that Ayurveda has built into its tenet. And as regards baseline Prakriti is concerned, Ayurveda talks very clearly about, say for example, if somebody has a Vat dominant, Pit dominant or Kapha dominant, it will manifest itself at different systems levels and give rise to structure and function related features like physical features, the metabolic features, the respiratory or activities, It will also manifest in the form of response to environment and stress, as well as that put together gives a susceptibility and progression to different conditions. So this is the framework that Ayurveda talks, that Vata, Pitta, Kapha are the physiological entities which carry out all functions, but their baseline proportions, they actually determine a lot how a person would in general have the built, and how the person will in general respond to the external environments. So this forms the basis for all the precision and predictive aspects in medicine. On the other hand, modern medical side or a modern genomics says that every individual has a unique genetic makeup, although more than 99.99% is something we shared, but the small number of variations that we have can actually translate into variations in how we express the genes, in the variations in how we metabolize the things, how we harbor the microbiome, what kind of physiological patterns it gives rise to. So in a way, it talks about Vata, Pitta, Kapha related manifestations which appear right, starting from the Shukra Shoni to the whole organism level. Here is modern science talking about multi-omic level differences which contribute to this kind of variability. The question was whether can there be can Prakriti have a genomic and molecular explanation, and if so, can it provide a window into the biology of Tridosha? So we set out way back in 2002 to look for molecular and genomic correlates underlying Prakriti, and thereby identifying the molecular correlates of dosha. And for that, what we needed to do is look at the diversity, stratify the normal healthy individuals into Prakriti groups, and look at their molecular and genomic profiles. So I will not get into those details in this talk because I'm trying to move to the how this kind of an understanding of molecular analysis of Vata, Pitta and Kapha through Prakriti can actually provide us to understanding of how molecular mechanism of drugs, which are also known to act through doshas, and how that can help us in understanding their application in the new diseases. So this is just to give you a premise that 7 Prakriti types we identified. Out of those, we looked at the Ekal Prakriti individuals to look for genetic variations underlying them. What we got is that normal healthy individuals, when of a very confined age group of the same geoclimatic and ethnic conditions, we could classify them into Prakriti groups. And when we did their genotype to genetic variations at expression level as well as biochemical variations and genetic level, DNA level variations, what we find was, one of the story that I'm going to complete today is, between Pitta and Kapha, there was a difference with respect to EGLN1 genetic variation. And this genetic variation was also visible at the gene expression level, and that was shown to have effect on how individuals will respond to hypoxia in high altitude conditions, because EGLN1 is a oxygen sensor gene. So for now, this much is enough for especially for students. EGLN1 being an oxygen sensor in the cell, if the genetic expression level differences are seen, and the differences are also seen in a particular single nucleotide polymorphism, it actually was shown to be important for how individuals would respond to hypoxic and high altitude conditions. Now, is it so that this variation would be important only if people were to travel to high altitude condition, or if that would also have any impact on the disease which involves hypoxic response? So in our lab, what we did was, through the knowledge of Ayurveda, we did Prakriti analysis, identified Ekal Prakriti individuals, stratified them, looked at the gene expression pattern as well as molecular network, identified a genetic variation, which was different between Pitta and Kapha, and the same was also different between people of high altitude pulmonary edema and natives, and this was assigned importance in high altitude adaptation. The paper was published in PNAS. The next study that was done in collaboration with our institute's another scientist, Dr. Anurag, what he did using in the mouse studies of asthma, he showed that inhibiting this gene and that exaggerates the formation of HIF1 alpha, which is an hypoxia inducible factor. It exaggerates the asthma and it impacts the asthma severity. So now this gene, which was different between Pitta and Kapha, not only has an impact on a high altitude responsiveness, but it can also have impact on asthma severity. Now we also have subsequently looked for other genetic variations along with EGLN1, and one of them would be in the von Willebrand factor, which is responsible for thrombotic outcomes, and those things will also determine the differential thrombotic outcomes in hypoxic conditions. What I would like to take you further is on the asthma aspect, which is hypoxic response in exaggerated hypoxic response in allergic asthma can produce severe asthma. And then the question came, if through linga or through the prakriti features, if we have come across an hypoxia axis to be differing at a molecular level, and the same has been also shown to be important in asthma severity, can there be a medicine which can act on this particular axis to reverse the hypoxic response and give rise to relief in the severe asthma level? So what we worked on was a Adatoda vasica, which is Vasa. Most of you would be aware about this medicine. We know that Ayurveda describes this use of this Adatoda vasica in several conditions, including infectious conditions and complex conditions, lung injury, even in Hridrog, Gulma, several places it has been described to be important. But as far as dosha balancing activity is concerned, it is known to balance kapha pitta, and it has a cooling effect. It is known to have bitter, so it is tikta, shita, gunatmak medicine. Now, if this was the medicine which is known to have kapha pitta balancing activity, and it is useful in conditions which has got several inflammatory as well as thrombotic outcomes, can we actually test them in the asthma? So this is what we did. We developed an acute mouse model of asthma and tested it for OVA challenge induced asthma, and we tested the Adatoda vasica. We also developed a severe asthma model where, along with the OVA challenge, we also inhibited specifically this EGLN1 to enhance the hypoxic response, and in that exaggerated hypoxic response where the asthma becomes much more severe and also does not respond very well to the steroids, if this medicine could be reversing. And if it does at a phenotype level, whether it does through modulating this hypoxia outcome, which was earlier shown to be related to pitta kapha perturbation. So what we all standard parameters that are used in modern asthma laboratory are all tested: physiological level, airway resistance, airway inflammation; at tissue level, airway remodeling; at cellular level, inflammatory cytokines. All of it was established that okay, the disease is established in the model, and then whether the effects are also visible for Adatoda vasica. So as we can see, the airway resistance is also reversed with the help of, so here this is OVA plus Adatoda vasica, OVA plus, so this black dot is actually bringing down the whole airway resistance. Now we also checked for all the other histopathological changes, including mucin and collagen deposition and inflammatory changes. So, and in addition to that, we also saw whether HIF1 alpha concentration was also brought down by Adathoda Vasika. So this tells that AV treatment rescues a severe asthma phenotype through HIF1 dependent manner. Now the question was that if hypoxia, it is known in literature that hypoxia also has an effect on mitochondrial morphology as well as mitochondrial functions, and these disturbed mitochondrial morphofunctions are also relevant in asthma, as has been shown earlier. Our question did not stop by looking at effect of Adathoda only on asthma features or on the hypoxia axis alone. We also went on to subsequently show whether hypoxia induced subsequent changes which are happening at the cellular level, whether those are also being altered by Adathoda Vasika. So we developed a cell line model of hypoxia, chemical induced hypoxia, and looked for the treatment effects of Adathoda Vasika. We checked for the mitochondrial function related effects using oxygen consumption rate, ATP production, and maximal respiration, etc. We clearly see that Adathoda Vasika was able to restore the mitochondrial oxygen consumption rate as is shown here, and we were also able to see the HIF1 alpha related effects in the cellular levels. We then wanted to see whether microscopically we can see the change in the mitochondrial morphological parameters such as networks, and it reverses the dysfunction which is induced by cellular hypoxic response. So these are like elongation, network, area of the mitochondria which are altered by because of the hypoxic chemical induced hypoxia, and the Adathoda Vasika was shown to restore that kind of a function. So this thing was published in journal, American Journal of Lung Cellular and Molecular Physiology. Then we further now that Pitta Kapha axis, which was shown to be hypoxia related, important in asthma, and the same Pitta Kapha balancing medicine, which is Adathoda Vasika, we could show its effect in the same asthma model. Now the question is, if hypoxia is such a central pathophysiological concept, and inflammation is such a pathophysiologically central to several things, we wanted to further identify whether other pathologies which involves hyperinflammation as well as hypoxia as a modulating agent, can this medicine which is reversing the effect of hypoxia can be also useful in other disease conditions. So this is where the importance of deconvoluting Ayurveda's Vata Pitta Kaph's physiology and mechanistic understanding in modern pathological language would enable us to explore more effects. So what we found was that we looked for other models, or like CLP ligation induced sepsis model, specific hypoxia induced thrombotic model, even in the bleomycin induced lung fibrosis model. All these mouse models of different pathologies were created, and we found that Adathoda Vasika was showing effect at both structural as well as cytokine levels, and even in the transcriptome analysis we could find. So we found that in the hemostasis model there was inhibition of HIF1 alpha and inhibition of the activated platelet count. At the lung fibrosis model, again we found inhibition of HIF1 alpha, TGF beta, reduction in the collagen deposition. In the sepsis model, we found reduction in IL-6, restoration of IFN gamma, reduction in lung inflammation and blood exudation, increase in the survival rate, which was comparable to the positive control. Then we also performed transcriptomic analysis, whole transcriptome analysis of the lung homogenate, which was treated with the mice treated with Adatoda vasica. We found very important pathways to get down regulated, like hypoxia, collagen biosynthesis, TGF-beta regulation. So even at the transcriptome level, gene expression level, we could find this very clearly, significantly differing. So then we also wanted to check whether these things which are important, like hemostasis, fibrosis, sepsis, these were all coming to be relevant for COVID-19, and so that's what we felt that these pathological hallmarks that were coming to be affected or altered by Adatoda vasica in individual mouse models. During the COVID, we realized that all these pathologies are actually becoming very, very relevant even in the COVID. So we further went on to look for specifically, these are like host response to COVID disease. But if I was to look for specific antiviral response, we will have to perform antiviral inhibition assays. And before that, we wanted to see if there were molecular docking analysis also provides any leads. So these are the compounds which are present in the extract that we use. This is Adatoda vasica whole water soluble extract that we use throughout in our models. And these were the compounds which were present in them, and we did the molecular docking analysis. As you would see, in addition to the host genes, the antiviral viral genes were also being good docking score. So so these are like examples of host genes which are having a good docking score against the molecules, and we then looked for direct inhibition of SARS-CoV-2 virus in the vero cell lines by Adatoda vasica whole water soluble extract. We found significant decrease in the percentage inhibition was more than 63. So that's what prompted us to look for its multidimensional effects in COVID-19, starting from viral replication or viral entry related to cytokine storms, and at different severe stages, this seems to be having a combined effect both at the viral and as an antiviral as well as in the host response modulation. So this paper we also published in respiratory research. Adatoda vasica attenuates inflammatory and hypoxic response in preclinical mouse models, potential for repurposing in COVID-19. So this told that if you were to identify, understand the molecular basis of Vat, Pitta and Kapha and assign the importance of that molecular difference to some disease condition or to some environmental response condition, and if we were to pinpointedly, precisely identify medicines which can be useful for those molecular axis of Vat, Pitta and Kapha, we can actually understand the molecular mechanism which can let us do more repurposing with evidence and with a more clear explainability. So next, we have also looked at other drugs which are another thing that Ayurveda has unique is about many relationship between diseases and drugs. So one drug can be useful in many diseases, and one disease can have many drugs to prescribe. So there are several disease gene networks are also being appreciated even in modern science. So one of the medicines that we have also worked on is called Patha, which is Cisampilus pereira, which has been described in two classes according to Charak Samhita, which is Tanyashodhan as well as Jwarahara. And this tells about it being useful in hormonal disorders, lactation related disorders, as well as some things like fever. Now this looks very, very very, very diverse condition according to modern science. On one hand, it is a hormonal disorder, on the other, it is a viral condition. So how does one reconcile these two? And for the Ayurveda physicians as well as students, I would like to just remind you that stanya is also a rasa upadhatu, and jvara is also a disease of rasa dhatu. And there is something common that it must be working on rasagni, or that might be giving rise to different kind of applications of the same medicine. So we tried to look for, it's a multi-component herbal formulation. Can we probe disease gene connections? And if we can probe this disease gene connections by using other Sesempelus pereira, can we also then understand its molecular mechanism, and that will again repurpose it for, allow us to repurpose it for different kinds of conditions. So this is what was done using cell line studies. Sesempelus pereira was MCF7 cell lines were treated with Sesempelus pereira, and the whole genome gene expression profile was looked for. And using this data, we also mapped it with the connectivity map database and tried to look for matching signals from other drugs related signatures, and we could find that it was matching with the antiviral, some of the antiviral drugs and small compounds. And we then also looked for subsequent experiments, also led us to identify link between hormonal axis, estrogen receptor 1, as well as antiviral activity. So this tells that novel molecular biological links can also be discovered if we do a systematic exploration of Ayurvedic herbs, which are described to have this kind of plural kind of applications. So this is also published in scientific reports, and so this is just to give you a glimpse about translational potential that can come from systematic analysis of Trisutra Ayurveda combined with the modern advanced technologies, and not only generating your own data, but your data can be mapped to the global data to make more acceptable and identifiable associations from those things. So this is a summary of all the kind of systemic analysis of Prakriti related things that we do in our lab, where we do population stratification by Prakriti through anatomical, physiological, psychological and other attributes, classify individuals, identify groups which are homogeneous within themselves. And we have also developed machine learning based algorithm for Prakriti stratification, and we look for underlying molecular differences. These differences we also studied at different levels, both at microbiome level. We have also looked at genomic level, at the gene expression level, and what we identify is that hypoxia, hemostasis, drug response, immunoinflammatory markers, they vary between extreme Prakriti groups, and in a way they become like a marker for different dosha. What we also want to check is that whether the molecular differences that we see, they are also visible at a physiological level as to whether skin physiology or airway physiology or heart rate variability. At these system level also the differences can be seen amongst healthy individuals, although they are all healthy, but within the healthy different Prakriti, whether they exhibit differences in anthropometric parameters, in airway physiology parameters like lung function. So these are the example, these are the studies that we carry out in our Ayurgenomics Center of Excellence and Ayurgenomics unit at CSR IGIB. We also want to study, we also study at an individual cellular level whether the Prakritis can be having some differences in their basic biological processes. So we reported differences in the baseline cell proliferation rate amongst different Prakriti individuals. even in the cellular level, and that also corresponds to the difference in which they respond to UV radiation. So these kind of stress response studies are done on the lymphoblastoid cell lines, immortalized cell lines developed out of extreme Prakriti individuals. And we also do physiological profiling, as I said. So cellular level, physiological level, molecular level, Prakriti differences is what we are trying to study. And whatever difference we find, whether those difference has got any implication in the way people respond to environment, the way people develop disease, or the way people's disease trajectories can have, or how it can help in deciding the line of treatment. So that is the applied aspect of all these eugenomic studies that we conduct over here. And one example I shared with you about the these things leading to development of newer understanding of medicine and repurposing of them. So in summary, I would just like to, I think I've already spoken in summary, but this is just to tell that Ayurgenomics framework can lead to identification of molecular basis of Tridosha concept, which could lead to discovery of biomarkers as well as understanding new disease through shared pathological features. And the Aushadha axis also has a potential for repurposing through explainability, such as finding new application of old medicines for new disease by finding common pathological features targeted by them. Novel biological links can also be uncovered through systematic exploration. With this, I thank you all for your attention. This is our IGIB building, and we are housed in Mathura Road campus. And I would like to acknowledge all our collaborators, PIs, different PIs, and the students. We have a PhD program in Ayurgenomics at CSIR IGIB, where students from diverse disciplines actually come together for solving the questions of Ayurgenomics. We have multidisciplinary collaboration, starting from disease biologists to experimental biologists to molecular biologist and computational biologist, and we had extensive collaboration with Dr. Anurag's lab. I and Mitali started this whole Ayurgenomics under the mentorship of Professor Brahmachari, the then director of IGIB. And we have students. Asthma work was mainly led by Atish, and then subsequently Ritu. We have also conducted a clinical trial on COVID-19 with Adathoda Vasika. So this is something, and funding support from CSIR, Ministry of Ayush, is also duly acknowledged. So this is our past and present group, interdisciplinary group of students, PIs, collaborators, and we have a very, very energetic team at IGIB. And with this, I thank you all for your attention once again.
Thank you, ma'am. Aapka presentation was fine, no doubt, because you have got beautiful knowledge from this Ayurveda and modern also, and well compared to your study design. And most of all, we find that the paper indicates to the development of Ayurvedic concepts to the modern genomics, so to say Ayurgenomics. And the genetic variations, what you have described in the paper, that was completely on Prakriti, which is also indicated from our classical text. So far, the Basaka you have told that's a multi-dimensional function, and already it has been told in Bhavaprakas, Basayang Vidyamanayang Asayang Jibitasya cha Rakta Pitta Khaya Kasi Kimarthang Nabasidati. There is a challenge. It was a challenge by the author, ki if you are having this Basaka or Adathoda Vashika in your garden, you forget about Swasa, Kasha, or even blood vomiting. So you see how potency they are, and you have well also taken his trial in COVID-19 also, because post-COVID complications include mostly in throat infections and also upper respiratory tract infections. And finally also you have correlated this Ayurvedic Rasha Guna and all the things with your study. One important thing you have told that is predictive, which is also in Ayurveda, apart from this preventive and curative, that is called as in our Ayurveda Arishta and some such things of that is Indriya Sthan in Charaka Samhita, it is well described. The problem is that when we go to inside this particular text, then only we can find all the things you have incorporated in your paper. Arishta, there also you can predict certain disease in future to come, as it was being told by previous author, as artificial intelligence that is well documented in Charaka Samhita Arishtathana. Thank you, ma'am. Well presented, and it will be a bone milestone for us. Thank you once again. Namaskar.
Once again, a huge round of applause for our speakers. Now, I request our session chairs to please conclude.
Dr. Gopalji, thank you for taking care of most of my responsibilities. I think this was one of the very enlightening sessions that we have, very much in line with the theme of the conference that we are having. Fortunately, I know most of them, and I can tell that they have given the nearly 2.5 to 3 decades of experience has been crystallized here and presented to you, and I think you are very fortunate to have that. We went from the Ayurvedic biology to Professor Valiyathan's theory and then to Prakriti, followed by Ayur soft, followed by taking asthma as a model, getting into the network pharmacology as a reverse pharmacology and what not we are knowing. The Chinese invented that. Reverse pharmacology was invented by Japanese and we have been following it. And then we came to the rasayana and artificial intelligence use and ghee. So if you see, Professor Kalpana covered a very wide range of activities that are possible and they have a potent future also. When it comes to Professor Asmita Vele, she concentrated only one particular aspect on the neurocognitive functions. Her concentration was on a specific ghee-oriented product and what are the possible methods by which you can evaluate the cognitive functions and the effect of not only ghee, but also the different kinds of ingredients that go into that. Thereafter, we had an excellent presentation from Dr. Prithviraj. He represents a typical westernized approach where he talked about the herbal genomics. And he showed us the various samples of the carrots that they have. We are now interpreting and understanding the newer and newer concepts of microbiome in the form of microbiota, the good and bad. We are still struggling to find out ways of handling the whole concept of microbiome in the context of system biology, in the context of cellular and then genetic approaches. We are still struggling. We are trying to find out ways how to do that. If that is what is going to be needed, then the question is that which model we are going to adapt. So before I come to that, my comment, and Dr. Bhavna Parashar, and I know she and Mithali Mukherjee have been working in this area seriously for the last many years. But very interestingly, what is observed, that it started from the Vata Pitta Kapha or Tridosha Prakriti. But finally, in order to be more practical, they have to come out with a sample of a disease or a drug. So either you come to Aradhota Varsika, or you come to ghee, or you come to something. So where you look at a model and a therapeutic intervention to reverse through how the product is acting, how the ingredient is acting, how the pathway is coming. So this gives us a challenge, that how to encompass this huge jungle of modern sciences with the simplicity of Ayurveda. One thing that clearly touches me and which I have been proponent of it right from the beginning is we must adhere to the fundamental principles of Ayurveda, the Siddhantas. In many of these cases, we have begun with Samanya. Prakriti is Samanya. But when it happens, the disturbance, that is where the dosha comes. The word dosha, why was the word dosha used in the context of functionality? If the functional systems are there and the functional systems are variable, how are you going to chase it? Yesterday morning we had rains, Somebody who moved from Mumbai to Dehradun and then Haridwar had to face, and my body had to undergo the changes. I had food changes in my body. Now, how are we going to catch it and how are we going to chase it? So here is the method. We need a challenge of adaptability to technologies. And I think we need to evolve simpler based on Ayurveda, because Ayurveda has capacity to provide simpler solutions to that. That is where the challenge of integration, the challenge of adaptability to sciences will come in front of us. Just to make a joke before I retire, I have about 100 students here. Let us get those 5 samples, Anuragji. Let us get those 5 kinds of simple kinds of carrots, which I saw 5 colors very prominently. Make them a blind in a dark room. Ask them to taste. Take one raw material which is cleaned properly, take another which is cooked. Let's have a sample study the way the cosmetic people, the nutraceutical people, the way the food people do, those are into sensual studies, you are aware about it. And I am ready to challenge that many of our Ayurvedic students, those who are well-versed, they will be able to come to right conclusions about what carrot is going to use and what could be used. This could be a very simple experiment that we can do. हम जो रस गुण वीर्य विपाक की जो बात करते हैं, उसको सिंपलर प्रोबेबिलिटी से हम सोल्यूशन कर सकते हैं. ये एंड नहीं है, ये बिगिनिंग है, वो आपको आगे जाने का रास्ता दिखाएगा. क्योंकि हम यदि जंगल में जाएंगे तो हमें हमारी पेडेस्ट्रियल जो रोड नहीं मिलने वाला है. मेरा एक पेपर है, मैंने बार बार बात नहीं करता हूँ लेकिन मैं रह नहीं सकता हूँ. जब क्लिनिकल रिसर्च की बात हुई थी 10-12 साल पहले, मैंने एक पेपर लिखा था, मैपिंग द फॉरेस्ट और काउंटिंग द ट्रीज. सो योर रिसर्च मॉडलिटी, whether it is validation, whether it is clinical research, whether it is drug research, whether it is product development, whether it is quality control, or whether it is standardization, I think we have to use the technology for the purpose that we want to use it. So let technology be the tool. Let technology not be your sadhya. What we have been doing is, most of the time we are using the technology as sadhya. No, technology is a tool. And if you want to use the technology in the right manner, I think we have to have a right prameya. आपके सिद्धांत क्या है, आपकी अपेक्षा क्या है, आपका प्रयोजन क्या है, वो आप तय कीजिए, आपको टेक्नोलॉजी यूज करेंगे. थैंक यू वेरी मच.
फिर भी सब तो कुछ सर ने बोल दिया. खैर कुछ बात नहीं, लेकिन आज का जो मेन सबसे बड़ा मुद्दा था, और इस आयुर्जेनोमिक्स की आवश्यकता आज भी है. और आज से अगर हम इस जेनोमिक्स के ऊपर स्टडी जैसे शुरू हो गया है, उसको हमने पाठ्यक्रम में या बच्चों को जैसे कि कुछ सुविधाएं प्रोजेक्ट के बारे में पहले से अंडरस्टैंड उसको करना चाहिए, तो आगे जाके अनुसंधान में बहुत ही काम पे आएगा. और थैंक यू. वेद के कोश को जो बंद पड़ा हुआ है उसको खोलने की आवश्यकता है. एक एग्जाम्पल दूंगा मैं मेरे जीवन में, कि जब मैंने BAMS पास किया, डायरेक्टर एंड द प्रिंसिपल एंड द सुपरिटेंडेंट वो तीनों बैठे थे. एक ही क्वेश्चन डायरेक्टर ने पूछा. इट इज़ माई एक्सपीरियंस ऑफ 79 व्हेन आई वाज पोस्टेड एज मेडिकल अफसर. तो उन्होंने पूछा कि हमें कुछ नहीं चाहिए, आयुर्वेद पढ़ के आए हो, ये बताओ, एक ही ड्रग बताओ जो कि आज डिस्कस हो रही थी वासंग के ऊपर, बासा थोड़ा बासिका पर में, कि ऐसा एक ड्रग है ट्यूबरकुलोसिस को अच्छा कर सकता है, हर्बल प्लांट सिंगल है, और उस शास्त्रीय प्रमाण के साथ बोलो. क्योंकि उसमें बहुत ही प्रेशर देते थे हमारे क्लासिकल टेक्स्ट के ऊपर. तो हमने यही श्लोक बोला था, और बाय द गॉड इमीडिएटली मेरे को अपॉइंटमेंट ऑर्डर मिल गया. बासायांग विद्यमानायांग आसायांग जीवितस्य च रक्त पित्त क्षय काशी किमर्थं न वसितते. सिर्फ इतना नहीं, सुपरिटेंडेंट भी पूछा कि अच्छा ये बताओ ट्यूबरकुलोसिस होने से पहले क्या आप प्रेडिक्शन कैसे कर सकते हो. तो द सेम इंद्रिय स्थान से मैंने लिया कि सर वो एक पेशेंट ख्वाब देख सकता है, ड्रीम. यू मे इमेजिन इट दैट इज समथिंग दैट इज आउट ऑफ द टॉपिक. नो, समथिंग इन आवर क्लासिकल टेक्स्ट बुक चरक संहिता. तं नदीं बहायन्ति नदीर्विजलाश्च ते तो क्षमता क्षमतो क्षवनाप्यता, अर्थात वही लोग ट्यूबरक्लोसिस में हो सकते हैं अगर वो ख्वाब में कभी बंदर उनको बैठा के गाड़ी में ले जा रहा है एक नदी के पास जिसमें पानी नहीं है। और इस चीज़ को मैंने मेरे लास्ट 40 इयर्स ऑफ रिसर्च में मैंने ट्रायल किया। 2 ही केस मिला, टू दी ऑनेस्ट आई एम स्पीकिंग, 2 ही पेशेंट मेरे को मिले। हालांकि वो ट्यूबरक्लोसिस तो मल्टी ड्रग थेरेपी खाते थे, बट मेरे विचार से उनको कभी मैंने ये पूछा कि आपने इस तरह का कुछ ड्रीम देखे हैं ना? यस सर। एक बार एक बंदर मेरे को पकड़ पकड़ के नदी में ले जा रहा था। तो ये जो आयुर्वेद में लिखा गया समथिंग एल्स, इसको नजरअंदाज कभी मत कीजिएगा, ये मेरा आपसे सबसे विनम्र निवेदन। थैंक यू।
Thank you once again, sir, for concluding this session. Now, my request to Dr. Kalpana Joshi madam to please come on the stage, and Dr. Asmita Vele madam to please come on the stage for felicitation ceremony. Please give a huge round of applause to both of our speakers. My request to our session chairs to felicitate both of our dynamic speakers. Please keep applauding for them. Thank you, Kalpana madam. Please keep applauding for them. We are once again thankful to our speakers for such informative talk. Now, my request to Dr. Anurag Srivastav sir and Dr. Anurag Vashne sir to felicitate our honorable session chairs. My request to sir to please come on the stage for felicitation ceremony. Please welcome them with a huge round of applause. Please welcome our dignitaries with a huge round of applause. My request to our audience is to please keep applauding for them. We are once again thankful to our speakers and our session chairs. A special thank to Dr. Anurag Srivastav sir and Dr. Anurag Vashne sir. Now, next we have special talk by our phenomenal speaker, Dr. Savita Lochab madam. Madam is currently serving as senior scientist in Patanjali Research Institute. Her areas of research include deciphering mechanism of action of Ayurvedic formulations against several diseases, including cancer, vitiligo, etc. Her presence at both national and international platforms are noteworthy. Please welcome such a dynamic speaker with a huge round of applause.
ओम। परम पूज्य स्वामी जी, परम श्रद्धेय आचार्य जी, जैसे कि कल स्वामी जी ने बोला था कि आयुर्वेदिक मेडिसिन्स और फॉर्मुलेशंस में वो पोटेंशियल है कि हम उनको एज जो हम जब कोई डिजीज़ होता है या जो हम इमरजेंसी में जो मेडिसिन्स देते हैं तो क्या हम आयुर्वेदिक मेडिसिन्स को भी, आयुर्वेदिक फॉर्मुलेशंस को भी उस तरीके से पेशेंट्स को इमरजेंसी में दे सकते हैं कि नहीं है? तो मेरे साथ जितने भी साइंटिस्ट या जो लोग भी पतंजलि में काम कर रहे हैं at molecular and cellular level, so I would say that they all are going to agree to that yes, we can project Ayurvedic medicines in emergency situations as well, because if Ayurvedic medicines can be used for treating the complex diseases like cancer, then they can be definitely projected as emergency medicines. So, I am Dr. Savita Locher, part of Patanjali Research Foundation, and I would share few of the research projects which we are working at the cellular and the molecular levels of Ayurvedic medicines, and we are looking into the mechanism of action of these Ayurvedic medicines in combating cancer. So, if I talk about So if I talk about cancer, then the WHO Globicant report in 2020, it almost said that almost 10 million people died because of cancer. And the new cancer cases by 2040 are going to increase to 28.4 million. This is a huge amount, huge number of people who would be having cancer. So this huge number is also considering the fact that we would be having advancements in the clinical settings as well. But that also indicates that only the modern medicines or the allopathic medicines are not enough in combating this cancer. So we need to have a new developed mode of regime, or the treatment mode of regime that can be used to treat cancer. So we at Patanjali thought of exploiting the traditional medicinal system of developing a new medicinal approach towards these complex diseases. Now, generally, when if we look at the drug discovery route, so from the bench side to the bedside, this is a long route. It includes, there are a lot of years of around 15 to 20 years that are invested in developing a single drug, single molecule that can be given to the patients. Along with that, a lot of a humongous amount of cost that is associated with that. And in addition to that, the percentage of the failure is too high in the routine drug discovery route. So we at Patanjali believe in reverse pharmacology, where we inspire ourselves from the already prescribed Ayurvedic formulations from the Ayurvedacharyas, which is prescribed to the patients, and we use those formulations and then look into the molecular mechanism of actions, how these formulations are helpful in treating specific diseases. So we work towards the at the molecular level of understanding that. And once we identify those molecules, then we can project it as a single drug or the molecule that can be used for treating the patients. Now, how Patanjali is working towards this, towards the combating of cancer. So we have a systematic way of approaching towards a specific disease. So we are inspired by Ayurveda and we develop specific formulations. Then we have specific herbal and the analytical chemistry which deeply characterize these formulations. And after that, we have in vivo, in vitro and microbiology team that decipher the mechanism of actions of those individual phytometabolites that can be used against the specific disease. I would be talking about leukemia and lung cancer. Now, before proceeding towards leukemia, it is important to understand the composition of blood. Blood is majorly composed of plasma and the formed elements. These formed elements are nothing but the cells. So these cells are specific cells like erythrocytes, leukocytes, platelets, and these all cells perform their specific functions. So there is a specific hematopoietic pathway through which these cells are formed. They are hematopoietic stem cells which develop into the precursor cells, and depending on the need of the cell and depending on the growth factors and the transcription factors and protein levels, these precursor cells are getting transformed into getting the lineage and getting transformed into the specific cells. These then they become blood cells, platelets, macrophages, lymphocytes, etc. So what actually happens in leukemia is that this transformation from the precursor cells towards the specific cell lineage, this gets perturbed. Maybe because of the changes in the protein expression levels within the cell, that the environment of the cell doesn't get the expression levels of the specific transcription factors or specific growth factors, that doesn't allow the transformation of these precursor cells into the specific cell lineage. So this happens in leukemia. So this is the normal blood where we have specific cells, but in case of leukemia, these untransformed cells actually outnumber the lineage specific cells, and that is why blood doesn't perform its function. So we have tested Divyacystograde diamond, which is one of the formulation of Patanjali against leukemia. What is special about Divyacystograde diamond is its composition. It is composed of Hirak Bhasma, and in Ayurveda, Hirak Bhasma has been called as Mrityunjay, so something that is having the quality to defeat death. So we were curious to understand the mechanism of action of Divyacystograde diamond against leukemia. So in vitro biology, how we can test that a specific formulation is effective against a cell against a specific disease is we have cell line models. These are established cell lines which were once procured from the cancer leukemic patients, and then we culture these cells in the laboratory conditions. So this is K562 cell line. K562 is derived from chronic myelogenous leukemic patient, and we have treated these cells with Divyacystograde diamond. So these are the bright field images. The lower panel is the bright field images where we can see that the untreated cells, they are round, they are blast. The dark region in these cells indicate the nuclei, which is big, and the nuclear and cytoplasmic ratios are way high. But when we have treated these cells with Divyacystograde diamond, we could see that the morphology of the cells have increased. The cells have increased their number, the size, and even the dense part is getting reduced. That means the nuclear and the cytoplasmic ratio is getting changed. So something is happening with the treatment of Divyacystograde diamond. When we have stained these cells with Jimza, where we can clearly look at the morphology of the nuclear, and we found that there are serious nuclear changes which we can see, that the morphology of the nucleus is changing. That means that something is happening with the treatment of Divyacystograde diamond. Now these are the cells of HL60. These are the cells which were procured from acute myeloid leukemic patient, and again we can see that with the treatment of Divyacystograde diamond, the morphological changes are happening in HL60 cell line as well. And this is a Jimza stain where we can clearly see that the big nucleus, the morphological changes are happening with Divyacystograde diamond treatment. So what are these morphological changes? Now if I go back to my first slide and if I look, that there were few cells where these are neutrophils which have this kind of morphology. So if we compare this picture with the cells, the images which we have obtained, we can clearly match them, that the cells which were once blast, so these are blast cells which doesn't have a specific nuclear morphology, when treated with Divyacystograde diamond, they have sort of attained these morphologies. Now what are these morphologies? So how can we actually validate that yes, they have attained a specific lineage of a cell type in a blood? So how we can look into that? So whenever a cell attains a specific lineage, it is having surface markers, specific surface markers. Like in this case, in this cell, if this is having specific surface markers, these surface markers would not match with the other one. So we have tested this with a technology known as flow cytometry. So in that, using that flow cytometry technique, we can look at what is the percentage of the cell that has attained a specific lineage. So this is CD41 marker which we have looked. So this is the percentage of the blast cells where only 18.8% of the cells which were showing CD41 marker. So CD41 marker is a megakaryocytic lineage marker. So when we have treated them with divycystograde diamond, the percentage of the cells have showing CD41 marker has increased from 18% to 42%, which is a significant change. That means that yes, with divycystograde diamond, it is not only that we can look in the change of the morphology of the cells through eyes only, but yes, the percentage of a cell, the percentage of the cells which are attaining a specific lineage is also changing. So this is CD11B, which is myeloid differentiation marker, which is also changing from 8% to 17.9% in the cells which were treated with divycystograde diamond in K562 cells. Similarly, in HL60 cells which were treated with the divycystograde diamond, so the myeloid lineage marker, which indicates differentiation in the cells, this is changing from 27% to 43%, which is again a significant change. So this indicates that divycystograde diamond is definitely effective against the leukemic cell lines in the preclinical settings. Now our next aim towards this is to identify that can we use this as a differentiation therapy. So what happens in differentiation therapy is that in case of leukemia, either you treat the cells with a specific compound that is going to push these cells towards apoptosis, या तो वो ट्रीटमेंट उन सेल्स को मार दे या फिर उन सेल्स को किसी स्पेसिफिक लीनिएज के टूवर्ड्स पुश कर दे. तो हमने ये देखा है कि दिव्यसिस्टोग्रेड डायमंड के ट्रीटमेंट से जो ल्यूकीमिक सेल्स हैं वो एक डिफरेंशिएशन थेरेपी को फॉलो करता है. सेल्स मरती तो हैं कुछ परसेंटेज ऑफ सेल्स, बट ज़्यादातर सेल्स जो है वो एक स्पेसिफिक लीनिएज को अटेन करती हैं. सो वी आर टूवर्ड्स आइडेंटिफाइंग द मैकेनिज्म ऑफ एक्शन कि ये जो लीनिएज शिफ्ट हो रहा है वो किस मैकेनिज्म के थ्रू हो रहा है. एंड द नेक्स्ट स्टेप इज गोइंग टू बी द पेशेंट सैम्पल्स in which we are going to procure the PBMCs from the patients, and we are going to try treating those cells in the in vitro settings. We are going to treat them with the divycystograde diamond, and we are going to look how cells, whether the cells which are procured from the patients are also undergoing any kind of differentiation. Now the next part of this presentation is lung cancer. We have also been working on lung cancer, and I'm talking about a single herb that is Pistacia antigerrima, जिसको आयुर्वेद और संस्कृत में कर्कट श्रृंगी बोलते हैं. तो आयुर्वेद में कर्कट श्रृंगी का जो यूज़ है वो रेस्पिरेटरी डिसऑर्डर्स के लिए किया गया है. So what we have, we got inspired with that information, and we have tested Karkata Shringi Pistacia antigerrima against lung cancer. So this is a big study. So I've just I'm going to only show you the schematic how we have approached towards identifying the role of Pistacia antigerrima in treating lung cancer. So these were the galls of Pistacia antigerrima. इसका हमने हाइड्रोमिथेनॉलिक एक्सट्रैक्ट जो है A549 सेल्स, वो सेल्स अगेन जो कि एक बार कभी हमने किसी लंग कैंसर पेशेंट से प्रोक्योर करी थी और अब हम उनको इन विट्रो लैब्स के अंदर ग्रो कर रहे हैं. सो वी हैव ट्रीटेड दोज A549 लंग कैंसर सेल्स विद Pistacia antigerrima, एंड वी हैव सीन कि उसके ट्रीटमेंट से जो सेल्स हैं वो मर रही हैं. अब वो जो मर रही है वो किस मैकेनिज्म से मर रही है, तो वो हमने टेस्ट किया है. दैट सेल्स आर डाइंग बिकॉज ऑफ एपोप्टोसिस. एपोप्टोसिस के अंदर भी वो किस तरीके से एपोप्टोसिस की तरफ पुश हो रही हैं. So there was, we observed that there is an exaggerated autophagy that is happening, and because of that exaggerated autophagy, cells are getting pushed towards apoptosis. We have looked into the molecular level as well, जहाँ पर AMPK phosphorylate हो रहा है, phospho-AMPK. So these are all the protein level changes which we have observed, and because of the changes in these protein levels, cells are moving towards apoptosis through exaggerated autophagy. Not only this, but we have parallelly what we have done is that we have also identified the specific phytochemical which is present in Pistacia antigerrima that is responsible for the anti-lung cancer property of Pistacia antigerrima. So we took help from the herbal chemistry team, and there were 40 fractions of the hydromethanolic extract of Pistacia antigerrima. And out of these 40 fractions, we pooled them into 5 fractions based on their TLC pattern, and those 5 fractions were individually tested against the lung cancer cells. And we identified that it was penta-O-galoyl beta-D-glucose, which was majorly responsible for the anti-cancer properties of Pistacia antigerrima. So this study has already been published in Frontiers in Pharmacology, but we have not stopped here. We have also working towards how Pistacia antigerrima is at a high throughput level is modulating the protein, the gene expression levels in the lung cancer cells. And we have identified that there are a lot of cells, lot of genes which were related or associated with the cell migration, invasion, assays, apoptosis, autophagy, and cell division. So, and not only this, we have identified a lot of genes which were specifically regulating the metaphase to anaphase transition of a cell. So, whenever there is a, in case of cancer cell division, जब होता है, so there are a lot of steps through which a cell divides. So we have identified that it is the Pistacia antigerrima is specifically targeting a specific point of cell division जहाँ पर है. तो शायद आयुर्वेद मेडिसिन के अंदर इतना डीप मैकेनिज्म ऑफ एक्शन शायद ही किसी ने दिखाया है, जहाँ पर हम लोग ये देख सकते हैं कि एक पूरे सेल साइकिल के पाथवे के अंदर भी मेटाफेज टू एनाफेज ट्रांजिशन में हमने देखा है कि मैक्सिमम जीन्स जो हमें मिल रही हैं वो इस पाथवे को टारगेट कर रही हैं और इसको इनहिबिट कर रहा है. So this study is ongoing. So we are working towards this. So thank you very much, and I would like to acknowledge the infrastructure and the facilities by Swamiji and Acharya ji. So we are able to work we are provided with the high-end technologies, and we are able to identify the mechanism of action of these herbal formulations. Thank you.
Thank you, madam. Thank you for illuminating us regarding this vital concept. Please give a huge round of applause to madam. Now, I request Dr. Jaya Balan sir to please come on the stage to felicitate such a dynamic speaker. My request to Dr. Savita Lochab madam to please come on the stage. Savita madam, please come on the stage. Please give a huge round of applause to her. Please keep applauding for her. Thank you once again, madam and sir. Now, it's time for lunch, and the arrangements have been made in University Mess. Please be back by 2:15 for the next session.